Hello.
Hello. Hello.
The meeting will start in 6 minutes.
Okay, thank you. Should I start now?
Oh, we can start now.
Okay. Should I start now?
Yes, you can. We can share you. We can see your presentation.
Okay, do you see my presentation?
Yes, we can see.
Okay, thank you. I'll start now. Hello, I am Seojun Lee from Gyeonggi Science High School in South Korea. Today, I will introduce you to my research about explaining the behavior of the electrical conductivity of a graphite line on paper using percolation theory. It is a well-known fact that graphite is an excellent conductor. However, when you draw a line on paper using a pencil and connect a multimeter to it, you often find that current cannot flow through the line at all. Why? When you draw a graphite line on paper and magnify it, you will find that microscopic graphite particles are stuck between crevices on the surface of the paper. This means that the graphite is not a continuous bulk, but rather it is an inhomogeneous system of particles, which may or may not be in contact with each other. In order for the graphite line to be conducting, there must be at least one conducting path that connects the graphite particles. For example, in this case, electricity cannot be conducting even though there are graphite particles on the paper. Now, this is where percolation theory comes in. Percolation theory predicts the electrical properties of systems like this by treating them as graphs and using statistical methods. In percolation theory, the fraction of all space that the conductive material takes up, which is called the filling fraction, determines the electrical conductivity. At first, when the filling fraction is low, there are no available conductive paths. This makes the system an insulator. But as the filling fraction increases, there is a specific point where the conductivity abruptly increases. This point, as you can see, is called the critical point. Quantitatively, the conductivity near the critical point is predicted by percolation theory to follow a power law as shown. Here, sigma is the conductivity, V is the filling fraction, K is a scaling factor, Vc is the critical filling fraction, and T is the critical exponent. The scaling factor, the critical filling fraction, and the critical exponent are constant parameters. Out of these, Vc and T are especially important because Vc shows you where the critical point is, and T shows you how fast the conductivity increases at the critical point. To examine the correlation between the filling fraction and the conductivity, I chose two variables to change in my experiments. First, I changed the number of iterations that the line was drawn, because more graphite particles will be deposited as you draw the line multiple times over the same location. Second, I changed the pressure that was applied on the graphite while drawing the line. In order to be able to consistently draw a graphite line under the same conditions, I designed an experimental apparatus. As you can see, the graphite is attached to a vertical column that is free to move, and weights are placed on top to vary the pressure. The paper is pulled out sideways from underneath the table. This is the picture of the actual apparatus that I built. Before we go on to the experiment part, I defined a new variable that can express the macroscale characteristics of the line more adequately. Because the filling fraction of graphite becomes sparser as you go deeper into the paper, I define eta to be the inverse of the resistance per length of the line, and measured eta instead of the conductivity in my experiments. Here is my experiment process. In order to express eta as a function of the filling fraction, I measured eta and v each as a function of the number of iterations, n. Then I expressed eta as a function of v by parametrizing both of them with n. This process was repeated for different pressure conditions. For my first experiment, where I measure eta while changing the number of iterations, I used the 4-probe method to measure the resistance of the line. Using a simple ohm meter yields large errors because the contact resistance between the probes and the graphite line is very large. The 4-probe method uses 4 probes instead of 2, which are positioned with equal spacing on the line. This method can mitigate the problem of contact resistance. The resistance per unit length of the line was measured as shown as the function of the number of iterations. 5 specimens were taken for each case. You can see that the resistance decreases with n, and that it also decreases with the pressure applied. The inverse of the resistance per unit length, eta, was calculated from the previous data. Points where the resistance could not be measured were treated as having an eta value of 0. Clearly, eta increases with the number of iterations. In my second experiment, I measured the filling fraction while varying the number of iterations. First, to measure the filling fraction, I analyzed images of the graphite line taken using a microscope. First, the images were converted to grayscale, and then they were binarized using Otsu's method, which is a statistical method that can calculate the best threshold between white and black. The results were as shown. Each point represents the average of 3 specimens. As the number of iterations was increased, the filling fraction increased quickly at first, but then it plateaued to a constant value around 0.8. In order to parametrize eta and the filling fraction v with the number of iterations n, a smooth and increasing relation between v and n was required. I constructed an empirical ansatz, as shown, using a probabilistic approach. Fitting this ansatz onto experimental data yielded high R-squared values of at least 0.97. Using the data gathered until now, I expressed the inverse of the resistance per length, eta, as a function of the filling fraction v. The critical point is easily seen at a threshold around 0.75 for all 3 pressure conditions. I fitted the theoretical power law function to my data and got R-squared values of at least 0.97. This means that the behavior of the graphite line follows percolation theory quite well. The scaling factor K had a clear positive correlation with pressure. However, the critical filling fraction and critical exponent values were almost unaffected by the pressure, as evidenced by the very small errors. The critical filling fraction had an average of 0.751 with a standard error of 0.0047, and the critical exponent had an average of 1.925 with a standard error of 0.013. So, here are my conclusions. First, a graphite line on paper was experimentally confirmed to be an electrical percolation system. Second, the critical filling fraction and critical exponent were shown to be almost unaffected by the pressure. This means that the critical values are not dependent on how the line is drawn. One possible application of an electrically percolating graphite line is that of inexpensive strain sensors. When a graphite line is prepared so that it is very near the percolation threshold, the resistance of the line becomes very sensitive to the curvature of the paper. For example, when the paper is curved away from the graphite, as in the second diagram, the distance between the particles increases, making the conductivity decrease. The converse happens when you curve the paper towards the line. This simple device can be made very easily with low cost. Also, drawing the graphite line does not need a separate apparatus, and it can be done by hand because it was measured that the percolation threshold is almost unaffected by the pressure. Therefore, this device may possibly be used as appropriate technology in third world countries. Here are my references. Thank you for listening to my presentation.
Thank you for your presentation. Do our experts have any questions?
Yes, thank you for your presentation. Can you please told us again about practical application of your work, of your results?
The practical applications.
Yes.
Okay, so I believe that a graphite line drawn on paper can be used as a very cheap strain sensor, so if you make a graphite line so that it is near the percolation threshold, then the resistance of the line will change very rapidly when you change the curvature of the paper by curving it like in the diagram. So yeah, so if you paste the paper onto an object and then you make strains in the object, then you can measure those strains by measuring the resistance of the line.
You have another experiments with another material, not paper.
I have not used another material, and I have only used paper. However, I know that there are other researchers who have tried using other materials, such as polymer, and I think those are quite interesting, but they are not in the scope of my research.
Thank you for your presentation. I have one question. Did you use only one type of pencil, or did you use different ones?
Because I wanted to control the type of graphite that was used, I used a pure graphite pencil that came from the brand Faber-Castell, and I think that a little bit different results can be achieved if you use different types of pencils from other companies. However, yes, so I hope that if I have the chance, I want to experiment with other types of pencils.
Okay, I see. But maybe can you suppose how the conductivity of the line depends on the hardness of the pencil?
So, well, actual pencil lead is not pure graphite, and it has a little bit of clay added into it in order to make the pencil lead softer. So as you can see, softer lead has more clay put into it, which means that the line will be less conductive because of the clay, which is non-conducting. So I believe that the conductivity will increase as the hardness increases because there is less clay put into harder pencils.
Thank you very much for your answers. Thanks.
Do our experts have any other questions?
Thank you. We don't have any other questions. We can go to the next.
Okay, we are waiting for our next participant. Taisia Simenova from Russia. She is connected now.
Thank you for listening to my presentation, and I hope you all have a great time today.
Thank you.
Thank you.
Our participant has connected. Taisia, can you hear us?
Yeah, I can.
You can start.
Oh, thank you. A second, please. I'll put here my demo. Can you see it? Just.
Yep.
Oh, okay. So my name is Simenova Tessa. I'm really glad to meet you, everyone there. And most important, my project's name is the study of air bubbles in a column of liquid in case of a non-inertial reference frame, or to be brief, sinking bubbles. Let me show you the object of the study first. It is a problem from International Young Physicists Tournament of 2019, which reads that when a container of liquid, for example, water, oscillates vertically, it is possible that bubbles of the liquid move downwards instead of rising. And I should have investigated this phenomenon. And actually, I was particularly curious about this phenomenon as it defies the common sense observation of bubbles always floating. Here you can see the demonstration. Moreover, if we talk about its practical usage, we could try finding its application in industry, which is especially relevant economical branch for our region, Yakutia. I guess you might even know Apple Diamond as a clear example. So it can be used in foam flotation, which is based on the interaction between basically water, mineral pulp, and bubbles with themselves. Therefore, my aim is to study this phenomenon. Here we go. Our first objective was to gather some information on its problem topic. We've analyzed a set of scientific and educational literature where we found a beautiful mathematical model of the sinking bubbles. You can see it here. It implies that while the vessel oscillates parallel to gravity, there exists a specific depth at which bubbles will remain motionless. However, this state is unstable, and the stable solutions are when bubbles form below or above this critical depth and rising and sinking occur. The mathematical model is based on such laws of nature as bubble volume with the application of ideal gas law and liquid vibrations, which leads to oscillating pressure is formula below and at the left side, where P initial is the pressure outside the liquid and rho x part is the increased pressure as a function of depth. This can be expanded into another question with the assumption that the Laplace pressure greatly overwhelms the forces acting on the bubble. Plus, there is Archimedes' principle, which states that the upward force of an submerged object is equal to the weight of the fluid displaced by it. And this arises from a difference in pressure between the upper and lower ends of the body of fluids. The lower end will have a higher pressure and will accelerate the body upwards. And after combining the various forces associated with the motion and inserting the model parameters into the Newton's second law, the governing equation of the bubble system is given by this formula. It consists of drag force that is determined by the cross-sectional area and the velocity of the object in the fluid attached mass. This means that the bubbles moving through a liquid will need to displace the fluid around it to occupy a new position. And this means that during its motion, the object must dispense energy to move its own mass and accelerate the fluid around its bulk. So this section must increase the kinetic energy of the water, and the object must do additional work to accomplish it. And actually, this way of considering the problem is only complicated when creating a model. So they used a computer simulation there. Sorry, I'm saying the attached mass is caused by a spherical object moving through a liquid, and it is half mass of the fluid displaced by it. As a common method in harmonic analysis, the method of separation of variables is used. And this separation uses the equation of general motion, like a fast motion, for instance, where the bubbles move upward, downward, or it's just stationary. And, oh, sorry, I was talking about the slow motion. Okay, and now we go to the fast motion. So it describes the fast oscillating terms in the model. Since the fast time scale function C is periodic, it has an average value of 0 over 1 period. And on the other hand, the slow time scale functions has an average value of itself. Therefore, the whole trajectory of the bubble has an average value. So we just use the function xt. Rayleigh integration is to integrate Newton's equation of motion. It is frequently used to calculate trajectories of particles in molecular dynamic simulations and computer graphics. In the paper that we analyzed, in order to observe the states of air in the vessel, they created a computational model that utilized Rayleigh integration to produce solutions. And in order to understand the stability of the regimes, we can plot the values of omega. You can see it on the scheme, x initial and the final depth xf, sinking given by a small enough initial depth. You can see now that the oscillation of fluids will only allow the bubbles to sink if specific conditions are met. We've created an experimental setup from scratch. You could have seen it before. It consists of a transparent vessel that is fixed on a speaker to which electric current is applied from a generator. And these QRs below lead to some more demos of the setup. Here you can see the observation table that shows how the bubbles condition changes with frequency control. And typically, bubbles arise at 30 Hertz and reach the bottom at 70. And in conclusion, the experimental setup created by us proves the mathematical model provided by analysis of scientific literature. And that means that the bubbles sink when the induced mass overcomes the buoyancy of the gas-saturated liquid. the immersion of the bubbles is affected by the frequency and amplitude of the wave. And in addition, principle of the sinking bubbles can be considered as a reverse flotation in mirror processing. From sort of curiosity, I tried adding oil into the vessel, and you can see here how it works. I hope so. Oh yeah, you can see the vegetable oil in the upper part, and water somehow takes it from this layer with itself. I think that's all. I'm open to answer your questions.
Thank you for your presentations. Do our experts have any questions? Anyone?
Thank you for your presentation. I have one question. Can you please remind us what is the meaning of x in your equations?
Minimum of x?
The meaning of x.
The meaning. Wait a minute. Oh, it wasn't needed. Sorry. You mean this slide where there was a table?
Yes, yes, with x more than x0, x less, etc.
Uh-huh.
Can you show us again the slide with this parameter?
Yeah, I'm trying to. Yeah, you. Oh. So,
Yes, that's fine.
and the usual x is like sorry, I need to look through my text to find it. It may take a time. Probably I couldn't answer this question because we don't have that much time to look through the whole theory I've used. So that will be fine. I'm sorry.
Okay, then.
I'm gonna work on it. The fact is that we just took the theory from the analyzed scientific and educational literature. So, oh my God, I can find it.
Okay, you don't have to answer this. If it takes so much time, we can move on.
Okay, we have 5 more minutes, but if our participant from China, Zhang Yu, is ready, we can start.
Okay, goodbye. Thank you.
Right.
Hi.
Hello, you can start.
Hi. Okay. Hi everyone. I am Zhang Yue from Qingdao No. 2 Middle School of Shandong Province, China. I'm very glad to have this chance to share my works with you. The title of my project is Screening of Tumor Drug Resistance Genes by Bioinformatics, EGFR targeting drugs as an example. This is my outline. First, why I do this work? When I was a little boy, I loved animals. I loved them because they are beautiful, amazing, and full of changes. I appreciate the beauty of nature and the variety of species. When I entered the lab for the first time and watched under the microscope, I saw this, just the cells of lung cancer. And as you know, cancer has become a major threat to human health, killing millions of people per year. At that time, I began to realize that life is not just about its beauty. Life scientists must take responsibility to protect the beauty of life. This figure is from Cancer Statistics 2020. It can be found that the death rates for the lung, colorectal, breast, prostate cancer declines, and the declines for lung cancer accelerated. However, lung cancer still caused more deaths in 2017 than breast, prostate, colorectal, and brain cancers combined. EGFR, epidermal growth factor receptor, is a transmembrane glycoprotein. It belongs to a family of 4 related tyrosine kinase receptors and acts as a key mediator in cells signaling pathways involving cell proliferation, apoptosis, angiogenesis, and metastatic spread. The overexpression of EGFR in a variety of human tumors and the critical role of the EGFR in the cell signaling pathways makes it a primary target in cancer therapy. EGFR TKIs such as gefitinib showed high significant sensitivity for the treatment of patients with EGFR mutations such as L858R. And they are recommended by international guidelines as a first-line treatment regarding the higher efficacy and safety. Now, there are 4 generations of EGFR TKIs that are used in first-line treatment. Despite the excellent disease control with initial EGFR TKI therapy, acquired resistance is ubiquitous and remains a key challenge. The first generation of EGFR TKIs, such as gefitinib and erlotinib, exhibit positive clinical responses between 50 and 80% of the patients who are responding. These patients with EGFR mutations. However, these patients who are responding to these drugs have faced secondary drug resistance after only 1 year of treatment, dramatically limiting the efficacy. The EGFR gatekeeper T790M mutation, also known as a primary mechanism of the drug resistance against first and second generation of EGFR TKIs, led to the discovery and the development of third generation of TKIs. Soon, new mutations such as C797S happened, and new TKIs have been introduced, and the 4th generations of EGFR TKIs have been introduced. Actually, multiple pathways have the potential to blunt efficacy and cause drug resistance. The multi-target agents and combination therapies are 2 appealing mechanisms to both overcome diverse resistance and enhance the efficacy of EGFR TKIs monotherapy. Molecular response profiling and drug screening should be used to investigate early adaptive responses after EGFR inhibition and identify candidate EGFR TKI escape mechanisms and combination therapy targets. In this paper, Differential genes associated with drug resistance to javitinim were screened by bioinformatics methods, and we hope to provide some help for exploring the candidate EGFR TKI escape mechanisms and finding the new combination therapy targets. Second, methods and results. 36 samples of non-small cell lung cancer obtained from GEO database were divided into treatment and non-treatment, drug resistance and non-resistant group. And the GEO2R was used to extract the differential expression genes. For the data from the accession GSE 75309, when the logarithm of fold change on the base 2 is larger than 1, it is chosen as a threshold criteria. 33 genes such as SIDE2, TAGLN have been found. For the data from the accession GSE 83666 with the same criteria, 86 genes, including SIDE2, LOX, GOP, KRT17, and so on, have been found. Notably, the gene SIDE2 show high difference expression between both treatment and non-treatment, drug resistance and non-resistant group, which may indicate its special significance in the study of javitininib resistance. SIDE2 is ubiquitously expressed protein exhibiting a high affinity for the CH1 domain of the transcriptional co-activator CBP-P300, also known as TAZ1. The CH1 domain of P300 also binds hypoxia-inducible factors 1 alpha, also known as HIF1 alpha, and some other transcription factors. Due to the interactions with CBP-P300, SIDE2 was reported to act both as a positive and a negative regulator of transcription. It appears to depend on whether SIDE2 enables the interactions between CBP-P300 and other transcription factors, or it prevents such interactions. The interactions between HIF1 alpha and TAZ1 under hypoxia conditions are related to transcriptional regulation of genes that are crucial for cancer cell survival. The role of SIDE2 in the inhibition of HIF1 alpha dependent transcription and the maintenance of cancer stem cells reveals its potential as a target in NSCLC. Post-drug candidates have tried to interpret the cancer cell survival mode by stopping HIF1 alpha from ever reaching TAZ1, but they haven't been very effective. In recent work, scientists found that it may be more efficient to mimic nature's own response and use a SIDE2-like drug to knock HIF1 alpha away. Despite the evidence linking both SIDE2 and HIF function independently to several aspects of cancer, little data linking this interplay between these two factors is available. Therefore, comprehensive studies are needed to develop innovative targeted therapies. 3. Conclusions and future. Based on the screening of differential expression genes, some possible therapeutic targets such as SIDE2 for the treatment of cancer were proposed. And as cancer cells continue to mutate, we have to continue to develop new drugs. It is like a circle without an ending. So I'm thinking that should we find some new ways to get out of this circle? A SIDE2-like drug or new combination therapy targets should be considered to develop for catching up with the speed of cancer cell mutation, in my opinion. Of course, further studies such as screening or more examples or new methods and the experiments should be done in the future. Thanks for listening. Thank you.
Hello.
Hello.
It's a very interesting theme. Thank you for this project. Say me, please, what software did you use for screening?
The software. The first, I just used some database. The first database I used was the GDSC database. On that database, I found that EGFR is very frequent in cancer mutations. So I think I chose EGFR as my target. The second database I used was the GEO database. On that database, I got two accessions. GSE 75309 and GSE 8366. I divided genes into different groups and used GO2R to extract the differential expression genes and got my results.
Okay, thank you. But maybe you use programs for modeling biological activity and toxicity compounds. It may be important for especially for synthetic chemists. No, don't use.
Yes. Excuse me, would you please ask the question again?
What program do you know for modeling biological activity and toxicity for compounds?
No problem. Problems.
Maybe PASS or QSAR. Don't use.
I think some problems. I think some problems is that the EGFR TKIs is very effective, but the drug resistance will eventually developed after nearly 1 year of treatment. That not only did it dramatically limiting its efficiency, and we have to continue to develop new drugs. So that this will make the drugs of these TKIs very high. So and makes many patients cannot afford this. So I think maybe we should find some new ways or use some combination therapy targets to treat this cancer. Maybe we can get better results.
Okay, thank you. It is
Thank you.
very big research. Thank you.
Thank you.
I agree with David. It's quite a large research, and I have one question. Can you please tell us just in 2 words, what is your own contribution to this work? Do you understand my question?
Yes. I think that I think I used the bioinformatic methods. Because of the COVID-19 epidemic, I have no chance to go to the laboratory to do any research. So I just used the bioinformatics methods to to find some data find some data and divided them into groups and extract the differential expression genes and got my results.
Okay, I see. Thank you for your answer.
Okay, thank you for your questions. Any more questions? Are they? Well, I propose to move to our next participant from Indonesia. There are two participants, Rizka Kurniawati and Jayan Shipshavati. You can start. Thank you.
Bye-bye. Bye-bye. Thank you. Bye-bye.
Hello.
I'm sorry, we're from Indonesia, but there's a technical inconvenience, so I need to log in because I used two devices, so I need to log in first. I'm sorry.
Okay, we have the time.
I'm sorry, could you please make me the panelist because um, I used two devices actually. Okay, thank you.
Okay, I added your account.
Okay, may I start?
Yes, you can. We can start.
Okay, Assalamualaikum warahmatullahi wabarakatuh, and good evening from Indonesia. It's such an honor for us to deliver our presentation in this incredible activity. My name is Dian Setiawati, and my partner, Rizka Kurniawati. Here, we would like to present you our presentation with the title Measurements of Red, Green, Blue or RGB Image Value, Light Intensity, Wave Value of Leaves, and Soil Salinity as the basis of health ecosystem mapping of Rhizopora stellosa. So, Indonesia is a country that known as Indonesia is a country that known as the Ring of Fire, which indicates that a lot of inevitable natural disaster could happen in the country, and one of them is tsunami. So on 28th of September 2018, in Palu City, there is a huge earthquake that followed by tsunami, sweeping shoreline houses and buildings on its way. And according to BNPB 2018, 2,256 people died because of this incident, and around 13.82 trillion rupiah as the result of the damage and the catastrophic losses. This indicates how crucial it is to maintain disaster mitigation in coastal area, especially mangrove, as a natural tsunami barrier. The conservation, restoration, and creation of mangrove forests may be one of the solution as many of the areas vulnerable to tsunamis and also as a home to mangrove forests. Moreover, an estimated 64% of the world's mangrove forests are within 25 kilometers of large urban centers and are threatened by future urban development, according to their 2014. And due to that, we think that it's important for us as the young generation to protect mangrove and give a contribution in the natural science research. So based on the, so moving on to the introduction. So the geographical condition of Palu Bay contains mangrove with most species of Rhizophora stylosa, grave or known as bangko. Based on previous research, this species is a mangrove species that can selectively overcome salinity levels, that is able to avoid salt absorption from growing media, and has the ability to adapt to extreme environmental conditions, such as soil conditions that are inundated with high salt content, according to Parit Muzaki 2012. Soil has different levels of salinity, depending on species and variations in each age level, and is influenced by other environmental factors such as temperature and so on. It should also be noted that Banaus village, which is the place for our research, is near with the urban area of Palu, so it will affect soil salinity based on salt content. Previous research states that increased salt levels can also be caused by changes in land use into residential areas, urban areas, and road networks. And also, soil salinity has a stable amount of salt content in it because it has a little effect on freshwater supply and rainwater. Water salinity has a significant role in mangrove restoration. The presence of salinity also has an effect on the leaves that secrete the salt glands or deviate, then subverted. The opinion about the effect of salinity states that in general, transpiration of mangrove species is low, while the roots continuously absorb salt water. This causes the accumulation of salt in the leaves. Salt accumulation in the leaves results in discoloration of the leaves due to different photosynthesis process. Rhizophora stylosa leaves have a light green to dark green. Previous research states that the Hue value is the color produced by the pigments in the leaves. According to Muhammad Saiful Adib in 2014, the Hue value is used to distinguish colors and determine redness, greenness, and blueness from light. The colors in the image have a range of scales that are different from each other, and the dominant values of red, green, and blue, or RGB. So the greener the color of the leaves from a mangrove, the mangrove can become a health bioindicator of mangroves. So Rhizophora stylosa adaptation process as the bioindicator by measuring soil salinity and color on the leaves through the image value of RGB with control light intensity to obtain Hue values. Next will be explained by my friends.
Let's move to research methods. This study used quantitative research method by observing the ability of Rhizophora stylosa as a mangrove plant by measuring the level of soil salinity and through the average value of the dominant RGB, control light intensity and uniformity, and analyze of leaf value through an analytical descriptive approach. The technique of determining Rhizophora stylosa as a sample, we plot the artificial area with the radius of each plot area of 10 meters by 10 meters box shape with the length of each side is 10 meters. For each plot area, we took 2 sample plots for radius 1 as far as 0 until 10 meters, radius 2 10 until 20 meters, and radius 3 for 20 until 30 meters from the shoreline. We will have 4 points for one plot area, and we give the radial rope as a divider. Next slide, please. Here we can see the pictures when we take data in the field. In the left pictures show when measuring the height of mangrove trees, and the right picture show when we measuring the diameter of a mangrove trees. Next slide, please. In this slide, show the prevalence of Rhizophora stylosa in the research place and when we taking a sample data. The study was located in Talise Palu Beach, Indonesia, with the distributions of the Rhizophora stylosa community in the taking sample data. Okay, for the first step of the research method is measurement of soil salinity value Based on sampling of soil around the sample tree as much as 100 milli, we took soil sample from each sample tree from each position based on 4 cardinal directions with a distance from the tree trunks as far as 10 centimeters. And next, measurement of RGB value on the leaf. The first step is to determine the sample leaf by counting the number of stems in one tree. Then in each stem, how many branches are counted. Then one branch as much as 10% of each branch. Then the leaf depth will be the sample. The second step, take a leaf color sample in the middle of the leaf using a camera with resolutions of 8 megapixel. The third step, get the RGB value by color analyze applications. Next slide. For the measuring variety of light intensity, when taking data, the RGB domain value requires control over uniformity of light. Taking light can be using loop methods. And the measurement of hue value on leaf and measurement analyze has conditions on the tendency of the dominant color, which is divided into red, green, and blue. To measure the hue value of a leaf, it will tend to measure the hue value, which has the predominant green color condition using this formula based on the pictures. The relationship between RGB value and the diversity of light intensity and hue value in a sample of one tree based on the observed leaves. At the end, we perform a correlation between the mean values, soil salinity, and hue value on our result and group it into a unit parameter by 3 groups, namely healthy mangrove, healthy enough mangrove, and mangrove unhealthy based on the value of the proportions of salinity tolerance. Next slide.
So moving on to the result. Here, first of all, we will give you the result of RGB value analysis. The research will take a photo in the middle or 1/3 of the leaf, the marked blue area. So the center of the leaf was chosen because it is the widest area of the leaf surface, so it is possible to get a more accurate color. And in here, from the graph shows that the value of red has a range, a minimum of 97 and a maximum of 143 with a mean of 106.8. Green has a minimum range of 139 and a maximum of 164 with a mean of 146.4. Blue has a minimum range of 58 and a maximum of 90 with an average of 65. And the RGB value is calculated using the color analyze. The average color based on the RGB leaves in 10 areas has a fundamental difference. So this is an indication that the leaf color can be related as a bioindicator. So the next slide is about the analysis of wave values and saturation. The wave values is used to render a color that is precise over the resulting RGB value. In finding the wave value, the researcher also gets the saturation value. The wave values are presented as follows. The wave value is used as a guideline that the light falling and forming a color is influenced by the angle from 0 to 360 degrees. Usually 0 is red, 60 degrees is yellow, 120 degrees is green, 180 degrees is cyan, 240 degrees is blue, and 300 degrees is a magenta. The wave value in 10 area has a mean of 89.1 with a minimum value of 77 and a maximum of 93. Of the 10 areas, the wave value has a standard deviation value, and this is for this STD for this shows that the data obtained is quite static, thus describing the conditions in research location that the wave value of Rhizoparus stilosa is uniform. Furthermore, Furthermore, the graph shows that the average saturation value of the 1,729 leaves spread over 10 mangrove areas is 54.7% and has a standard deviation of 0.39. This shows that the saturation value is even and has no significant difference for the change in the Hue value. Furthermore, research when taking RGB image will be influenced by the intensity of incoming light. Therefore, controlling the shooting distance as far as 50 centimeter with different intensity conditions shows that at the research location, the position of the leaves cannot always be directly exposed to strong sunlight. This is shown in the light intensity obtained during the data collection process. Different light intensities can be caused by the location of the leaf sample in different parts of the tree. And some location at a certain time get a strong light intensity. Some locations get a weak light intensity, and the light intensity values are follows in here. So light intensity analysis, research when taking RGB images will be influenced by the intensity of the incoming light. Moving on to soil salinity, pH, and conductivity analysis. So after measuring the values of RGB, Hue, and light intensity values, the research will then measure the water salinity value. The salinity of water is measured because it is an important factor that will affect the photosynthesis process. The need for plants in the photosynthesis process is water availability. However, in conditions in the mangrove forest, water availability will be related to the salt content it has. We took 100 milliliter sample of water in a 10 centimeter radius from the main stem of Rhizophora stylosa. The results of the salinity analysis for the 10 areas are presented as follows in here. So soil salinity in the 10 areas has an average of 2.4 ppt. As for the salinity in fresh water is less than 0.5 ppt and brackish water 0.5 ppt until 3 ppt. Meanwhile, seawater is above 3 ppt. At present, it is known that the salinity of water around the Rhizophora stylosa tree is categorized as a brackish water, and this is thought to be due to absorption by the stems and roots of Rhizophora stylosa, which can absorb excess salt levels. And analysis of pH and conductivity as measurement of soil salinity. So in contrast to water salinity, soil salinity is the salinity of solids, which must use a measure of electrical conductivity. To measure soil salinity, a conductivity value is needed and is measured using a conductivity. And as a four-valley groundwater salinity are as follows in here. From the table, it shows that the average value of soil salinity around Rhizophora stylosa is 0.744 microsiemen per centimeter. The low salinity value indicates an absorption of Rhizophora stylosa against the salt content in the soil surface. Rhizophora stylosa is able to select soil salinity levels. Soil conditions without acid and alkaline or neutral tend to be close to zero. And meanwhile, for brackish areas, the true soil salinity value is 4 until 10 microsiemen per centimeter. Difference proves the existence of the absorption process carried out by the Rhizophora stylosa. So next to the relation between the Rhizophora stylosa's adaptability to soil salinity, Hue value, true leaf RGB value, and light intensity. So utilization of mangroves can be used as a bioindicator of mangrove health. Some species have the ability to receive stimuli from changes in salt levels and other dissolved minerals. One of the potentials of mangrove plants can be seen in the Rhizophora stylosa. So the ability of Rhizophora stylosa to absorb salt and in plant respiration resulted in decreased water and soil salinity values. One hand, the presence of mangroves will provide benefits by desalination of seawater into fresh water. The Rhizophora stylosa mangrove undergoes a photosynthetic process by means of water being absorbed by the roots, and in this process, salt is absorbed with the water. Then it will give the leaves a color change, which tends to be bright in color. Under certain conditions, the Rhizophora stylosa will shed its leaf as a form of adaptation. And the last is potential of mangrove mapping as a bioindicator. So by understanding the mangrove morphometry of Rhizophora stylosa, especially leaf color based on RGB, Hue, and light intensity values, it can create a pattern of high salinity levels in a coastal area. If there is a change in color that is much brighter, it will be an indication of a higher salt content. And this indicates the salinity of the water, and the soil is influenced by the high salinity. So high salt content is an indicator of abnormal mangrove ecosystem. This is an early detection of damage to the coastline and can be addressed by revitalizing the coast. Low salt levels in the mangrove ecosystem have an impact on the inhibition of the photosynthesis process, and by minimum or potential limitation of mineral resources, abiotics in the soil, and changes in types or characteristics on the coast. So by understanding leaf morphometry, biochemical properties, conductivity, salinity, we can formulate and have a potential future research base to carry out an application. The foundation of this research will encourage the development of a color-based biodetector technology as a tool for mapping mangrove health. The importance of realizing mangrove health is to determine the potential, risk, and benefits of utilizing the adaptation properties of Rhizophora cellulosa as a bioindicator.
Okay, the conclusion in this study is that the measurement of soil salinity can be described by the identified color change with the average of RGB value. Red with 106.8, green 146.6, and blue 65.5, and the hue leaf value 89.1 at different light intensity levels of the Rhizophora stylosa plains. The relationship between soil salinity and RGB value through the calculation of the leaf hue of the Rhizophora stylosa plain as a bioindicator of mangrove health in the mangrove forest area can be used as a parameter and a solution for the ecosystem health map solution of Rhizophora stylosa. Can next, please? And the future plant, after knowing the basis of the green leaf color, the next research plan will be developed using the Arduino sensor to make a signal sending device for leaf color change as an adaptation of Rhizophora stylosa. This is the end of the presentations of our research result. Thank you for your great attention.
Thanks for your questions and for your answers. I propose to move to our next participant from China. Thank you for your presentation, Dan. Thanks, Rita.
Thank you.
Is our connected participant Ming Lei or Li Cheng? I'm sorry, I can't read it.
Yeah, we are here. Can you hear me?
Yeah, you can start.
Okay. Wait for a minute for the screen share.
Yes, yes, of course.
So, can you hear? Can you see the PowerPoint?
Yes, we can see your screen, but can you open the presentation in full screen?
We can see just a folder, not a presentation.
Camera. Yeah, is that okay now?
Yes.
Okay. I am Su Milei from high school affiliated to Shanghai Jiao Tong University.
So hi, I'm Lu Lichen from high school affiliated to Shanghai Jiao Tong University. So today we are going to talk about our project, Association of SLC6A3, ADR82A, COMT gene polymorphism with introversion and extroversion. This is our content, and our presentation mainly consists of 4 parts. Let's move into the first part, introduction. So the project is originated from one day I was chatting with Suming Lei, and we started wondering one question about how one's personality is affected, or what factors will affect one's personality. We think that is obviously related to his or her growth environment, and the education he or she received is also an important reason for the person's personality. But if there is any possibility that your personality is inherited from your parents, which is the problem we are wondering about, that if personality may be determined by your gene. So after we figure out the problem we want to discover, we found some problems comes after, like how do we determine one's personality, and like which gene should we choose to study? So bringing all these questions, we move into the part 2, methodology.
Okay, first of all, we need to know how do psychologists judge one's personality. And we find a very famous questionnaire called MBTI test on the internet, and it is divided into 4 dimensions. And in one of the dimensions, it talks about a person's introversion and extroversion personality. And we want to focus on this dimension because extroversion and introversion are popularly talked in our daily life, and it's very easy for us to understand how is the definition of extroversion and extroversion.
So then we start to design our questionnaire for introversion and extroversion. As Suming Lei just said, we use Myers-Briggs type indicator test, which is the most popular professional personality assessment tool in the world. And the concept of MBTI is raised by Carl Jung. He divided people's personality into 4 aspects, which the aspect we are focusing on is the extroversion and introversion. The aspect remain is like sensing, intuition, thinking, feeling, judging, and perceiving. So we choose the most conspicuous part of the personality, which is introversion and extroversion. So we find a reliable Chinese translation of MBTI's questionnaire. The original MBTI test consists 4 dimensions, and we choose and rearrange 21 questions for the part of introversion and extroversion from the 93 questions in the MBTI origin test. So this is our questionnaire design. It consists of 3 parts. The first part is a self-evaluation. In this part, participants are asked to choose words to describe themselves. And the second part, they are asked to choose the favorite word they like. And in the third part, the participants are asked to choose the actions they will take in a given situation. Among all these 21 questions, there are a honesty problem, which is the same question and same option with in reverse order. This is aimed to test whether the participants are treat this questionnaire seriously or not.
Next step is to choose which gene we are going to research on the association between them and the chosen personality, introversion and extroversion. And we find the first gene is SLC6A3, and it is a gene code for dopamine transporter. Dopamine is a neurotransmitter used to transport happiness and excitement. So we consider this gene has the possibility to relate it with introversion and extroversion. And the second gene we chose is ADRE2A. It is a adrenaline receptor gene, and it is secreted when adrenaline is secreted when people face urgent situations or get excited. So we consider this gene may also have the relationship with the personality. And the third gene is COMT gene, and it is code for an enzyme that breaks down dopamine and adrenaline, and was also said to have a relationship between 5 characteristics, including introversion and extroversion in the past researches. And so we also include this gene. this is our procedure to get the genotype. First of all, we need to get DNA from volunteers, and their oral epithelial cells are collected, and sodium chloride salting out method is used to get their gene from their cells. And the second step is to amplify the DNA. PCR is used in this step. And the third step is genotyping. Molecular beacon is used in this step. Molecular beacon detection is an analyzed technology developed based in two facts: resonance energy transfer phenomenon and the base complementary pairing principle. As a fluorescent labeled molecular probe has the following advantage: low background signal, high sensitivity, strong specific recognition, simple to operate, and real-time detection without separation from unreacted probes. The left table is the fluorescence waveform, and the next table is the genotype result judgment.
So now we move into the data analyze part. In this part, we mainly use two methods, which one is the Pearson chi-square test. We want to see if there's any association between single variables we choose and the introversion and extroversion. So we calculate the p-value. And also in the experiment, there are 4 classified variables, and personality and the 3 genotype. And these all variables are unordered, which is genotype like AA, AG, GG. So we use multi-nominal logistic regression methods to analyze it. It's a simple extension of binary logistic regression, which allows for more than 2 categories of the dependence or outcome variables to be analyzed. So now we got our conclusion. The research has 214 data samples, and their information is in the following table. And you can see the gender, you can see their grade, you can see their nation here to explore how different types of alleles on specific locus affecting the people's personality. We test 3 sites of the different genotypes, and the information is listed here. By counting the results of the subject, we contain the personality scores of the respondents, and we categorize the scores into 3 types. 30% of the respondents, they are introversion, and 40% of them are extroversion. According to all these statistical data, we can judge the influence of 5 variables of age, gender, ethnicity, grade, and genotype on extroversion and introversion personality. We got our result. Gender has significantly correlation with intro and extroversion personality. You can see its p-value is way much smaller than 0.05, which suits our hypothesis. After we figure out gender may have the correlation between extroversion and introversion, we also conduct the multivariate data analysis. We use multi-nominal logistic regression as previously said, and we got result. In this table, we can tell that CC genotype of RS1800544 was significantly correlated with extroversion, which is p-value smaller than 0.05. The result from the wild type and mutant type also proved our previous result. So RS1800544 is the locus of ADRA2A.
And it is the past 4 discussion part. In this project, we used an effective method to conduct genotyping, which is molecular beacon, and find that RS1800544 in ADRA2A has the association with introversion and extroversion. However, there are still some parts to improve because there are some controversy around the accuracy of the questionnaire, and the sample capacity is not big enough because there are only 214 volunteers.
This is the end of the presentation. Thanks for listening.
Okay, thank you for your presentations. we are going out of timetable, so our so questions have to be brief. Any questions, experts?
No.
Yes, I have one question. What is your own contribution to this work? Both.
So the question is, what is our contribution to this world?
Yes, to this project.
I think first of all, we discovered the mechanism between a psychological phenomenon and the its background information in biology, and it studying this mechanical, we can maybe further use the like the gene and the gene coded enzyme to cure for some psychological diseases, for example, depression or something that related to introversion and extroversion gene expression.
Maybe you don't mark. Just a moment. Okay, thank you.
Okay, thank for the questions. Thank you for your presentation. I propose to move to our next participants from Indonesia, Anjali Nuranda and Nabila Salsabila, if I spelled it correctly. Hello?
Hello.
You can start.
Okay, can you see the PowerPoint?
Yes, we can see.
Okay, thank you. Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello, the honorable juries and all the participants. We are so glad to be here and nice to meet you in this moment to the presentation day. Before we begin the presentation today, let us introduce ourselves. My name is Nabila Salsabila Daulay and my partner is Anjali Nuranda. We are from University of Bengkulu, Indonesia. Okay, in this moment, we want to presentation about our research, and the title is Hakakura, hand sanitizer, calamansi, Western skin, and aloe vera, antibacterial activities against Escherichia coli and Staphylococcus aureus bacteria. Okay, next. Okay, so this is our contents that will be explained today. First is background, second is problem formulations, third is research methods, fourth is results and discussions, and the last is conclusions. Okay, so I wanna tell you about our background. So Indonesia is increasing awareness of sanitation in the community. The average growth of sanitation development for a decade, 2006 until 2016, is 2% per year. However, in 2016, Indonesia has increased investment 40 times to 80% of sanitation development during 2016. One of the sanitation programs is the hand washing program, which is a simple activity that aims to remove dirt and minimize the number of germs on the hands and palms. One of the products that make it easier for humans to wash their hands is a hand sanitizer that contains antiseptic compounds consisting of alcohol and triclosan. Alcohol activity as an antimicrobial is effective at killing gram-positive and gram-negative bacteria, which can inhibit the growth of bacteria on the hands, such as Escherichia coli and Staphylococcus aureus bacteria. Hand sanitizers are antiseptic products used in improving simple sanitation programs, namely hand washing activities in Indonesia. But without realizing it, hand sanitizers can have a negative impact on health, such as Andrew in 2017 said, Hand sanitizers can increase levels of BPA in the urine and blood. Therefore, a hand sanitizer that comes from natural ingredients is needed, besides being safe for health. It also supports sanitation awareness in the community. After that is One Village, One Product, or OVOP, is one of the steps toward industrial clustering in the small and medium industry, or IKM, sector, which aims to promote regional superior products such as calamansi orange, as the first promote regional superior products such as model of the OVOP program in Bengkulu City in January 2011. And fruit skin calamansi orange contains essential oils, which consist of various components such as terpenes, sesquiterpenes, aldehydes, esters, and are antibacterial. As well as calamansi, the Bentara durian is also involved in the development of the local potential of the Bengkulu area. Thus, on BPS data, in 2012, the Bentara durian tree grows and is found in 10 district cities, and in 2011, the productions reached 331,16 quintal per year. Increased production of durian fruit in Bengkulu also affects the volume of waste that accumulates and decays. Durian peel is used as an antioxidant, anti-inflammatory, and contains secondary metabolite compounds that are antibacterial. And aloe vera has the potential to be developed as a medicinal and industrial raw material. Aloe vera has antimicrobial properties, namely anthraquinones and saponins, which can inhibit bacteria and fungi. So, according with the problems, we as researchers took the initiative to make hand sanitizers from calamansi extract, west durian skin, and aloe vera. Thus, the waste in the form of durian skin can be utilized more optimally. The product is declared as a breakthrough hand sanitizer from natural ingredients. Therefore, the researchers raised the title of the study, Hakakura, hand sanitizer, calamansi, west durian skin, and aloe vera, antibacterial activity against Escherichia coli and Staphylococcus aureus bacteria. Okay, can you next slide, please? Okay. So this is our problem formulations. First is knowing the procedure for making Hakakura. Second is knowing the antibacterial activity of Hakakura against Escherichia coli. Third is knowing the antibacterial activity of Hakakura against Staphylococcus aureus bacteria. And the last is knowing the antibacterial activity of Hakakura against Staphylococcus aureus bacteria. And our research benefit is first, make it easier for people to process waste that is difficult to process. Second is help develop the potential of Bengkulu province, especially in the field of agriculture and entrepreneurship. And the last is assisting the community in choosing a safe and environmentally friendly hand sanitizer. After that, the methods will be explained by my friends.
Okay, judges, I wanted to explain about research methods. The first is materials. This is time and place. This research was conducted from November 2019 to October 2020 at the research laboratory of the Faculty of Medicine and Health Sciences, Bengkulu University, Bengkulu City, Bengkulu Province, and material and tools. We use calamansi, durian skin, aloe vera, alcohol, newsprint, cotton, nutrient agar, pepper, and distilled water. And then we use research design is experimental method. We use purposive sampling, namely the selection of sample based on certain consideration obtained by researchers themselves on the characteristic and characterization of the known population. And I want to explain about steps. This is step to make Hakakura, hand sanitizer, calamansi, and durian skin. You can see this step. You can read this step in our slide. And these two steps to make hand sanitizer, you can see and you can read this step to make hand sanitizer. And this is the result. First, the antibacterial activity of Hakakura against Escherichia coli bacteria. The first extract, Restorzone, is 15.5% Escherichia coli, and positive control is 9.5% Escherichia coli. F0 is 8%, F1 9%, F2 13%, and the last F3 15%. 15%. And the second, I want to explain about the antibacterial activity of Sakura against Staphylococcus aureus bacteria. We use isolate Staphylococcus aureus. The first extract, Restorezone 16.5%, positive control is 10.5%, F0 8.7%, F1 12.5%, F2 is 15.5%, and the last F3 15.5%. And the last, the result of Sakura organoleptic observation in terms of questioning to customers, we use simple random sampling, and the result is first F0, shape is liquid, color no color, and smell is fragrant. And the second F1, shape is liquid, color is yellow, and the smell is fragrant. And the third F2, shape is liquid, color is yellow and greenish, and the smell is fragrant. And the last F3, shape liquid, color is brownish, and smell is fragrant. You can see it's the figure of clear zone test. Is the first figure one is clear zone test Escherichia coli. You can see this in our slide, and this in the last is figure 2 is clear zone test Staphylococcus aureus. And this is conclusion. The first, the alcohol extract of calamansi peel, Bentara durian was skin, and aloe vera with the maceration method has been shown to be able to inhibit the growth of Escherichia coli and Staphylococcus aureus bacteria very well, including those applied as hand sanitizer. And the second, hand sanitizer preparation for the extract of calamansi skin, visible durian skin, and aloe vera with a concentration of 0.5, 1, and 1.5%. Produce inhibition zones is Escherichia coli of 9, 13, and 15 millimeters. And the third is, well, Staphylococcus aureus produce inhibition zone of 12.5 millimeter, 15.5 millimeter, and 15.5 millimeter. The greater the extract concentration, the greater the inhibition zones. And the last is the quality of the hand sanitizer with the addition of calamansi skin extract, Bentara durian skin, and aloe vera past the organoleptic. The best hand sanitizer of the 4 formula is sanitizer is formula 3 because it produces the largest zone of inhibition and quality that still meets the quality standard of hand sanitizer. Okay, thank you very much. Thank you for your attention.
Thank you for your presentation. We have time for 1 or 2 brief questions.
Thank you for your report. I have one short question. How did you estimate the antibacterial activity of your Hakakura extract?
I'm sorry, pardon. Can you repeat your questions? I'm sorry, I don't hear that.
Yes, okay.
I'm sorry.
How did you estimate the antibacterial activity of your Hakakura extract? You showed us the tables with antibacterial activities. How did you get these numbers?
Okay, so we got the number is we testing in the laboratorium in the Faculty of Medicine first. After that, we using a method of macerations. After that is we using a simple random sampling for testing. So that's the reason we can get that number for the simple random sampling and testing in the laboratorium.
Okay, laboratory. Okay, thank you. I understand this. Thanks.
Thank you, the judges.
Okay. I propose to move to our next participant. just timetable. It's our next participant is Yang Zhao from China. Hello.
Hello, can you hear me?
Yeah, we can hear you. We can see your presentation. You can start.
Okay. Dear experts, good afternoon. I'm Zhao Yiyang from China. The topic of my chemical research is biomimetic synthesis of natural chromophoric phenoxy compounds. Let's begin the introduction through video presentation.
Hello, I'm Zhao Yiyang from Henan Experimental High School. My tutor is Professor Yang Guanyu from Zhengzhou University.
I fall in love with chemistry as early as 10 years old, and personally conduct basic experiments and collected 80 kinds of elements. I conducted chemical experiment activities at my home, independently get a variety of compounds. Also, I personally collected 80 kinds of chemical elements. In 2020, I grasped a precious opportunity to join in an InChai project. When Professor Yang told me about the project of biomimetic synthesis, it arouses my great enthusiasm. Without hesitation, I participated in it. Now, I feel it a privilege to participate in the International Online Forum of Scientific Youngs. Hope to communicate and make progress together in the forum. APXOs are widespread natural compounds, which was used as dye in ancient time and later developed as probes for bioimaging in modern days. APXO diversities are reported to demonstrate bioactivities to be used for the treatment of cancer, bacterial infection, and other diseases. Developing enzyme-mimicking catalytic models with a more cost-effective means of synthesis continues to be a great challenge. Sustainable technologies are still required with respect to environmental concerns, safety, cost, physical workup, and practical industrial application. Hurrying to pursue a green and sustainable enzyme-liking approach for synthesizing APXOs. A Lucky's mimicking model with natural gallic acid and manganese acetate was developed. As for the methods, I use ICF Finder and Web of Science to search and read literatures, ChemDraw and Origin Softs for drawing structures and reaction formula, catalytic impairments to study the catalytic for performance and optimize reaction conditions, reaction analysis with TLC and HPLC, purify our products by recrystallizations.
Yang, we can hear you. Yang. Hello. Yang, should it be any sound in your presentation? We can see your screen, but cannot hear you. Or sound. I have resent the instructions of how to share sound on Zoom. Should it be any sound in your video? Yiying.
Hear me now?
Yeah, we can hear you.
Can you hear this video?
The reactor was heated to 25.
It's good then.
Okay. May I replay this video again? Sorry.
Play from now. I don't think we missed too much. Play from now.
We are ready to continue watching your video.
Play from now.
Play from now. Okay.
And keep the pressure of oxygen and 0.3 megapascal for the deserved reaction time. The ending reaction mixture was acidated to pH 1 to 2. In the end, solid cake was washed with water for 3 times. To improve my comprehensive ability in practice, I assisted in group research and learned necessary knowledge. First, we conduct thermodynamism and achieved a yield of 99%. Then, we use instrument UV kinetics investigation and draw a curve of initial speed against the concentration, which confirms the measurement kinetics supporting a lattice mimicking behavior. My teacher told me this is a plausible mechanism of it. On top of that, we developed cross-color condensation with a yield of 98%. Through the above project research, I grasped the experimental skills and related knowledge. I wonder if it can process polycondensation, so I began my interest in several research intern. I conducted several tests to optimize the conditions and obtained a brown-rice-black compound with 70% yield. However, as special structure and possibilities, I still can't confirm its structure by HAMI and HPLC, but I find out its more clear weight in HRMS, in spite of about 0.198% error. So this means I got a target compound, and the synthesis method has its possibility and feasibility. I will conduct further research on polycalcification as soon as possible. To conclude, a green, convenience, and biomimetic method for the preparation of phenoxy compounds was developed. The demeritations occurred by using natural renewable gallic acid, manganese acetate, oxygen as oxidant, and water as solvent. The method with practical convenience and low loading of catalysts opens the door for future development. During my study and research, first, I learned lots of chemical experimental skills and knowledge. Second, I used an advanced method to conduct further study and achieve initial success. Next, I will continue my study and learn professional knowledge actively. I believe my research will get more success sooner or later.
Luckily, our project has published in journal Chemical Select recently. As for the reason why we choose this project, APXOs are widespread natural compounds which was used as dye in ancient time and later developed as probe for bioimaging in modern days. We developed enzyme mimicking catalytic models with the most cost-effective means of synthesis continue to be a great challenge. Hurrying to pursue a green and sustainable enzyme-lacking approach for synthesizing APXOs, we developed this lattice mimicking model. I use SCI Finder, Web of Science, ChemDraw, OriginSoft, catalytic experiments, TLC, HPLC, purifier plugs by recrystallization, NMR, HRMS, to carry out my research. And we process our reaction in a pressure tube with 2 aminophenol, gallic acid, manganese acetate, and water. The mixture was adjusted pH to 9. After the reactor closed, the mixture was changed with oxygen for 3 times. Then the reactor was heated to 25 degrees, and the pressure of oxygen under 0.3 megapascal. And then we got the solid acid, which was washed with water for 3 times. Thank you for your attention.
Thank you for your presentation. Questions? Dear experts, do you have any questions for this presentation?
Yes, I have one question. How did you track the progress of the reaction?
Pardon, could you repeat it?
Sorry, can you repeat?
Can you repeat your question again? Thank you.
How did you track the progress of the reaction?
You mean this one? Track the reaction. We use in-situ UV clinics investigation. And by recording the observe at
No, I mean thin layer chromatography or high effective chromatography.
Could you repeat it? I didn't hear it.
I mean thin layer chromatography or high effective chromatography.
You mean this one?
Um,
Uh,
no,
we
it's when you begin, you say that you used TLC, HPLC.
I know. We get the reaction liquid in the pressure tube and doing the TLC, and then after the reaction we use organic liquid to dissolve it and process HPLC to find out its details.
Oh, thank you. And what NMR spectra did you capture?
What numbers of which can you?
H13 or C13. NMR, nuclear magnetic resonance. What spectra did you use?
What you mean the type or?
No, NMR. Characterization of products by NMR. Yes, it is.
This?
Yes.
We use HMR.
H.
This picture.
Only H.
Yes. I began to learn it and use HMR to find out its structures.
Yes, it is very important for chemical synthesis.
Yes. And as for these products, we also conduct HRMS.
I'm sorry, we do not have much time.
Thank you.
The question is answered. Okay.
Oh, thank you for your question.
Thank you for your presentation. I propose to move to our next participant from Indonesia, Erlin Wijaya and Prajna Vijaya, if I spelled it correctly. Hello.
Hi everyone, I am Prashna Wijaya.
Hello, you can start.
And I am Ellen Wijaya.
Okay, may I share my screen now?
Yes.
Good afternoon, everyone. I am Prashna Wijaya, and that is my partner, Erlin Wijaya. And let us present our research titled EFU Package, an eco-friendly and practical pharmaceutical packaging design for elderly and children.
Medicines are used to treat diseases, manage conditions, and relieve symptoms. Around 94% of elderly around the world uses medicine. Marketed medicine packaging has several issues that elderly experience, like packaging too firm, unable to remove the medicine breaks when pushed through, cavity too small for medicine to go through, and many more. And personally, I think that this issue is also experienced by a lot of people in wider ages, that is not only elderly and children. The practical issues elderly experience is also similar to what many children experience in taking medicine. Medicine packaging, like blisters, are made up of plastics and aluminum foils that are usually disposed by landfilling, which caused by soil and incineration. These issues in medicine packaging might not be seen as a big deal, but in truth, practicability issues can harm children and elderly if they use inappropriate helping tools to take the medicine, for example, knife and cutter. And the environmental impact of pharmaceutical packaging is huge, since it is made up of 4% of total daily waste.
So based on the research background mentioned before, there are two research questions in this study. The first one is what materials that can be used to create a pharmaceutical packaging that is practical for elderly, children, and environmentally friendly, and how to create a pharmaceutical packaging design that is practical for elderly, children, and also environmentally friendly. The method used in this research is the LCA method, also known as the life cycle assessment method. It is started by assessing the environmental performance of total packaging by the review of gross material density, degree of filling, greenhouse gas emission, and finally the amount of product waste. Next would be the assessment of the total distribution cost of packed product, which would be the review of the cost of packaging materials, the cost of transport, and finally the cost of packaging loss. Afterwards, we did a task for the product quality and preservation, which is a literature research for each packaging materials. And lastly, we did a user friendliness test, which is done by the quantitative analysis of user friendliness by elderly aged more than 60 years old. So based on the LCA method, the result shows that based on the environmental performance of total packaging, the gross material density of polyethylene terephthalate is 1.38 times 10 to the power of 3 kg per meter cube. And the gross material density of biopack bioplastic is 1.29 times 10 to the power of 2 kg per meter cube. And the maximum degree of filling inside the biopack 3 times 4.5 centimeter cavity is 2 times 10 to the power of minus 6 meter cube. And what is so great about EVU package is that it is a zero waste product and it has a very low or even zero carbon emission. Next would be the total distribution cost of packed product. Since we are using the life cycle assessment method, every indicator in this research needs to be in the multiplication of 1,000. So 1,000 kilogram of package is equivalent to 1,984,126 packages, or it is equivalent to 152,981 US dollar, or approximately 8 cents in US dollar each. The price to ship 1,000 kilogram of package would be 430 US dollar if shipped by the Indonesian Post. Next will be the product quality and preservation. A biopack package can last for a year if stored in room temperature. And it is also resistant to air humidity, which means that the product inside won't be affected by air humidity. Moreover, PET is resistant to water and also gases. So the medicine package inside this EFU package would be resistant to air humidity, water, and also gases. And it will last for approximately a year. Lastly, for the user-friendliness test, according to 10 elderlies more than above 60 years old, EFU package is a practical packaging.
So these are the results of the PET packaging. As you can see, the left one is the closed or unsealed one, and the right side is the sealed one. The packaging is divided again into two parts, that is the inner and the outer part. The inner part is where you put the medicine, has 3 compartments. Next, this is the biopack inner sachet. So whenever you need to take your medicine, you just need to tear the red part, and the right side is the layout of the nut packaging. All these labels are based on World Health Organization Annex 9. A more practical or easier packaging design for elderly and children with low risk. EFPP is also a solution for land pollution and air pollution due to ammonium landfilling and incineration of pharmaceutical package. EFPP versus previous packaging. Blisters and strips are more economically effective than EFPP. However, they are made of plastics and ammoniums that can potentially harm the environment in the disposal. It has also several practicability issues. Next is bottle. It has several practicability issues that is unlikely to be managed by elderly and children, but on the other hand, they are more economically effective than EFPP.
So finally, to conclude this presentation, there are several material candidates that is suitable for pharmaceutical packaging. And out of all of it, the most suitable materials used in EFU package is the polyethylene terephthalate and biopack bioplastic. According to the data gained from the LCA method, the GMD of polyethylene terephthalate is 1.38 times 10 to the power of 3 kg per meter cube, and the gross material density of BioPak is 129 kg per meter cube. The degree of filling inside the BioPak 3 times 4.5 cavity in EFUV package is 2 times 10 to the power of -6 meter cube. And in the whole process of EFUV package making, there is no greenhouse gas emission. Instead, greenhouse gas is reduced in the process. Moreover, EFUV package is a zero waste package. The cost of 1,000 kilogram of package, or 1,984,126 packages, costs $152,981 or approximately 8 cents in US dollar each. To ship 1,000 kilogram of EFUV package from Indonesia by the Indonesian Post is $430 US. Since EFUV is zero waste, then there is no cost of packaging loss. According to the interview to elderly aged more than 60 years old, EFUV is also a practical packaging for elderly.
Future work. Many different adaptations, tests, and experiments have been left for the future due to COVID-19 pandemic currently happening. Future work concerns deeper analysis of particular mechanisms, new proposals to try different methods, or simply curiosity. The following ideas could be tested. 1. The future researches could be done to fulfill the data gaps in this research, preferably after this COVID-19 pandemic abated. Other forms or modified packaging that is more practical, sustainable, and simple can be developed.
A modified packaging from this packaging that is more practical, sustainable, and simple can also be developed. And lastly, a practicability trials with larger amount of participants and wider age group, maybe including children or even other age groups, could be done. And that will be the end of our presentation. Thank you. Are there any questions?
Yes, thank you for your presentation. Just one question. Do you already have any ideas of this modified packaging? Maybe you already created something.
We currently haven't yet, to be specific, sir.
Okay, thank you.
Thank you for your presentation. We have a time for one more question. 2 minutes. Are there any questions? Dear experts? Well, our next participant has connected, Rachel Maconier. Can you hear us? Hello?
Hello, everyone.
You can start.
So greetings to you, everyone. I'm Rachel Makonye from Queen Elizabeth School, Zimbabwe. My project is about a plastic dissolving capsule for livestock, which is a solution to ruminant impaction. So first of all, we need to define the term ruminant impaction. So ruminant impaction due to plastic is a condition in which indigestible plastic foreign bodies accumulate in the rumen of ruminants, leading to indigestion and many other adverse health effects. The most common plastic involved is usually polystyrene or styrofoam. So in developing countries, it is mostly used as it is cheap and as it is a cheap material. Mostly food vendors, especially in my country, Zimbabwe, mostly food vendors in the non-formal sector are usually the people who use this styrofoam, usually to cut costs and increase profits. Here I have examples of styrofoam projects. As you can see, there are K-lights and disposable containers, coffee containers. And here there is a food vendor, or also known as a food seller, who is packaging her food in a styrofoam container. So the problem is that once people eat this food in these packages, they litter the container, causing pollution, which is a very big problem to the environment. Let's take a look at the land pollution and animal health. More than 1.3 billion tons of plastic are being deposited on land worldwide. Some of it ends up in grazing lands or simply areas where livestock reside. Livestock then confuse it for food and end up ingesting the material. This plastic ends up being clogged and it affects digestive system, leading to a slow, painful death. Animals, being innocent victims of human careless behavior, have to suffer the side effects of plastic. Most animals in the extensive farming system are mostly affected. However, all the livestock are at risk because of the continuous rise in pollution. So this means that even animals in the intensive system can also be affected. So I found an article that says in 2017, a Times of India cited veterinary officials and animal welfare groups estimating that around 100 cows die annually just in the city of Lucknow from eating plastic. I also have a case in Nyanga. So Nyanga is a town in Zimbabwe. I once visited it and I discovered that in every 10 households, there were about 9 people who had their livestock dying due to plastic, and they have no solution in the meantime. I have, however, come with a solution to ruminal impaction. I've introduced the plastic dissolving capsule to help in this major problem. In short, you can just call it the PDC. It is the first ever invention that targets ruminal impaction. So through this, it will be through the use of this capsule pill. At the moment, only surgical methods are available, and of which they are expensive and they require special qualifications, and of which not all people can afford to go to a veterinary doctor. So the animals end up dying and losing their livestock. Here I have a procedure on how to make the gelatin capsule. So it is a hard gelatin kind of capsule made in a process called encapsulation. Here I have a definition of encapsulation. Encapsulation that thus refers to a range of dosage forms or techniques used to enclose medicine in a relatively stable shell known as a capsule. The following stages are involved in encapsulation. Step 1, we prepare the gelatin solution. So during the preparation of the gelatin solution, we dissolve gelatin in water at a temperature of 60 to 70 degrees Celsius. So once our gelatin has been melted, we look at the viscosity of the solution. So because our solution will be at 30 to 40% weight by weight, our solution will be highly viscous, meaning that some air bubbles are going to be formed as a result of air entrapment. So we use a vacuum to remove these air bubbles, as they can cause inconsistent shells at the end of the process. Step 2, we have the deep coating and deep coating the gelatin solution onto metal molds or pins. So during this process, we are molding the capsule shells. So we mold them in special chambers, also known that will be at a constant temperature of 50 degrees Celsius. Step 3, there's rotation of the deep coated pins. This is to evenly spread the solution onto the pins, and also this depends on the size of the capsules. Step 4, with the drying of the gelatin solution. So during drying, the metal pins will get their shape and they can then be further processed in step 5, which is stripping and trimming. So during stripping and trimming, we're removing the excess gelatin solution in order to retain our beautiful, or should I say, the perfect gelatin capsule. Step 6, we have the joining and trimming of the capsule shell. So this is whereby we join the two capsules. So during joining of the trimmed capsule shell, we have two parts and they need to be closed. So we use a procedure called a pre-locking system. And this then helps that, especially maybe in pharmacies, they need to then print the capsules in order for identification. So the capsules can then be joined, and then step 7 can be introduced, which is printing. Yes. So what are the major parameters? My major parameters in this project are viscosity, temperature, and pH levels. Viscosity. Here I'll be looking at the gelatin solution. So the viscosity of the gelatin solution should be at a constant viscosity, because if the solution is very viscous, then it means that our capsule shells won't be good enough. So we need to look at the viscosity of the shells. We also look at temperature. So temperature in the making of the gelatin solution. During the making of the gelatin solution, first we look at the constant temperature during which we dissolve the gelatin. So we need to keep the temperature at 60 to 70 degrees Celsius. And then during drying, we also look at temperature again. So our temperature needs to be 50 degrees Celsius. Okay, so we also look at temperature during the administration of the capsule. During the administration of the capsule, okay. So during the administration of the capsule, we are looking at the temperature inside the livestock animal. So we are looking especially in the rumen. So our temperature needs to be high in order for the capsule to break and let its contents out so that the chemical reactions can take place. pH levels. So pH levels will be looking at the pH levels during administration or during digestion. We are going to look to see that during the digestion of this capsule, the factor that is going to make the capsule to break is the high pH levels in the stomach of the animal. So the acidity in the animal's stomach is going to make this capsule to break in order for the chemical reactions to also take place. Here I have just a diagram representation of the encapsulation process. It is simply going to summarize the main processes that take place. So here is spinning, dipping, spinning, drying, stripping, cutting, and joining. Contents of the capsule. I have carbohydrate supplements. I have included them in my capsule because they will help recover energy that would have been lost due to the loss of appetite in the animal. So I'm simply saying that once the animal is suffering from ruminal impaction, it loses its appetite. And this loss of appetite then leads to the animal not eating and losing a large amount of energy. So the carbohydrate supplements are going to help. I've also included vitamin B1, which is also known as thiamine, and it helps body cells change carbohydrates into energy. And also it improves appetite. So vitamin B1 is a well-known nutrient that, that controls the hypothalamus part of the brain, which is in control of satiety. So thus it is going to help in improving appetite. So I also have acetone. Acetone is a well-known organic solvent that dissolves or breaks down plastic. Due to their similar properties, acetone can dissolve the high carbon hydrogen in styrofoam. Administration of the capsule. The capsule will be taken orally. So this means that it will be taken from the mouth and it will move down the esophagus until it reaches the rumen, where it will break and let its contents out due to the stomach's acidity, as I have said before in the major parameters. So since acetone is my main ingredient, it will act on the polystyrene, also known as styrofoam, in the rumen. And then the rest of the nutrient supplements will later be absorbed in the bloodstream. So once this plastic has been digested, it will be excreted as part of waste, and the nutrients will still remain in the body and they will help in the animal's health. So here I have done an experiment to test the breaking down of polystyrene in acetone. As you can see, I have my polystyrene and also acetone. And I have recognized that acetone is a very good solvent and it is going to break down this polystyrene into gooey substances. And thus it is going to help in the capsule. The observations and results. I observed the formation of air bubbles once the styrofoam was dipped in the acetone. It was sort of like boiling when I added the styrofoam into the acetone. And it took about only 20 seconds to dissolve a 15 by 2 centimeter block of polystyrene, which shows efficiency of the solvent acetone. The material turned from a solid to a slimy and also gooey substance. Resulting conclusion. Styrofoam consists mainly of air, and when it dissolves in acetone, it completely loses its structure. Due to their similar polarities, acetone can dissolve the carbon-hydrogen bonds in styrofoam. So dosage. Dosage is a very important factor in this capsule. So we need to look at the dosage chart because if we give the animal overdosage, the animal may suffer the side effects such as acetonemia, ketosis. So underdosage and overdosage may occur when the capsule is improperly taken. So you have to look at weight, age, species of the animals, and the species of the animal, as they play an important role during deciding the capsule sizes. Here I have a capsule chart. So let me take an example of a goat, an adult goat. So an adult goat would take a capsule size of 10. So capsule size of 10 is 10 to 18 grams. So it is going to be administered by the adult goats. So the capsule sizes will range from the age, as I've said before.
I'm sorry to interrupt, but you are running out of time. You have to speed up.
All right. So here are the future research and conclusions. After the production and administration of the capsule, animal health will be taken care of. Food security and biodiversity will also no longer be at stake. Livestock provide important economic and social roles for the well-being of communities and world at large, and therefore losing them results in a major loss. So here I'm simply saying that animals provide food, raw materials, and other stuff. So by using this capsule, we're ensuring food security and also other roles that they play. I, however, wish to improve my project in the near future to be able to solve ruminant impaction due to all the plastic, which will include polyethylene, PET, high density, HDPE. So, thank you.
Thank you for your presentation. Do you know that acetone is very dangerous for organism?
Yes.
But is it possible for cows and other animals to have acetone inside the organism?
So here's what happens. If the animal ingests too much of the acetone, then this means that there may be side effects. So
what is the dangerous concentration for cow, for example?
Okay, so if too much acetone is ingested, it may lead to acetonemia ketosis, which is a problem that will affect the milk yield of the animal. If it's a cow, for example, it will reduce the milk yield. So I have, that is why I've used the dosage chart to avoid overdosage of the acetone. So we need to use the dosage chart in order to avoid overdosage.
Would it be enough for plastic disappearance, such doses?
Sorry?
Would it be enough for plastic disappearance if you told about doses?
Okay. So yeah, the plastic won't be disappearing as such. It will be simply dissolving into a smaller material, which is easier to digest. Because here the problem is that there's a blockage in the rumen, which is affecting the digestion of the plastic. So this capsule is simply going to help dissolve, or let's say break down the plastic for it to be easily digested.
Okay, thank you.
I suppose we can move on to another.
Okay, thank you for the presentation and answers. We are now waiting for our Next participant, Francia.
Thank you.
Thank you, Aisha.
Thank you.
The participant from France should have connected 5 minutes ago. We are waiting now. Our participant has connected. Hello, Tristan. Can you hear us?
Hello, yes, I can hear you.
Yes, you can start.
So I'm gonna set the presentation. Can you see the presentation?
Yes.
Yes. I'm gonna set it up. All right, so hello everyone, I'm Tristan Rodeau from France. My team and I decided to work on the crystal growth. We've worked on it because in physics class, we discovered the crystals and we see those magnificent pictures where we can see big crystals in caves. And it's questioning us because we don't really know how does it work and how can we create such a magnificent thing. So crystals are all around us, like table salt, diamonds or snowflakes, and we don't really understand how does it work. So we managed to do first tests. So we make a solution crystallization. It was messy looking crystals, so we wanted to get the crystals that fit with their original structures. We decided to have a look and have a closer look on the primers. So it's where the crystals begins to grow. With this observation, we decided to choose the primers with 2 criteria. They has to be as big as possible, and they can't have twins because because the crystals are an accumulation of different layers of the chemical species, and if the original structure is messy, the structure of the final results will be also messy. So when we have choose our primers, we have to put it in the solution so that they can grow. We decided to at first to attach it with the hair, but it was really hard and meticulous. It took us a lot of time. So we decided to use another technique using a drilling system. So here's a quick view of the montage. So we use a rotary tool, which rotates a thick drill of 0.1 millimeter. The crystal is on the support. It's a microscope bottom so that we can precisely set the crystal up and down and know the size of our crystals. We mainly decided to work with copper sulfate. It was cheap, easy to use, and the crystals were very easy to have. The crystals are growing in solutions, so we have to know the exact concentration that we are using. If the concentration is too low, the growing time is very long, and so because we don't have this time, we manage to get another concentration. And if it's too high, the growing is very fast, and we can't control what is happening in the solution. Like it can crystallize very fastly, and so we can't obtain what we wanted. We decided to choose an average concentration, which is a perfect balance between the growing time and the quality of the crystal at the end. We've also decided to follow the beginning of the crystallization by kinetic monitoring. So we use another chemical species, potassium dihydrogen phosphate, which gives us flat crystals, easy to produce in rows. And we've sent it to a laboratory with an electronic microscope. So there is a picture of a crystal without a hole, and here is a crystal with a hole. When we send it, we wanted to know if the crystal has the drilling system affects the structure of the crystal. And as you can see on this picture, the internal structure of the crystal is not affected at all. So our drilling system were working well. And when we looked at the side of the crystals, we saw those little dents looking like two sawtooth on the sides, and it's where the chemical species was setting and make the crystal grow more and more. Here's a picture where the copper wire is going through the crystal. And here you can see the node. We wanted to know if an obstacle on the crystal were affecting the growing of it. And here you can see with the nose that the crystal covers up the node without modifying its structure. So it wasn't a problem anymore. Here you can see the crystal is filling up the hole, and it doesn't affect the form of the crystal. We also wanted to know the volumic mass of our crystals. We use spectrometers, but as the crystals are soluble in water, we have to work with alcohol. So we have to know the volumic mass or the density of alcohol. And then we apply the technique with our crystals in alcohol and without. We find with the copper sulfate density very close to the reference, so our technique were working well, and we were able to do it with other chemical species. And when the crystal was growing, we've decided to measure the mass through time to have more data about the growing time. So here's a quick view of our system. As we know, the mass given by the balance, it was easy to know the real mass of the crystal through time. With the push of Archimedes and the weights, we were able to know the exact mass of the crystal through time. We also wanted to know if other parameters such as temperature or humidity in the room was affecting the growing of the crystal. So we use Arduino with a sensor that gives us the temperature and the humidity in the room through time. And all this data was sent to the PC where we can draw graphs, etc., etc. And our final experiment, we recently tried to reduce the concentration in the solution. And as the crystal is affected by water, we have to use ethanol. So because the ethanol is, because when alcohol is dropped in water, the solution warms up and the crystal dissolves. So we have to use a perfusion to add drip by drip the alcohol. So those experiments led us to a nice evolution. Like we were going from crystals to 1 centimeters to 15 to 20 centimeters. And they were looking as close as the perfection that we were searching for.
Thank you for your presentation. Any questions from our experts?
Yes, thank you for your presentation. I have a few questions. Firstly, can you please explain what is the name of your crystal?
I've done
Goldsmith crystal. What is it?
Goldsmith, in French, it's orfèvrerie. It's like the job of men's which manipulates precious resources such as gold or I don't remember the word in English, but it's a work where people manipulate very
Jewelry, yes?
Excuse me?
Jewelry.
Yes. So it was the name because we have lots of parameters to know to control the growing time of our crystals, and it's required very precise measurements, and yes, that's
Okay,
what.
I see. Yes, and what is the purpose of your work? Can you please?
At first, the purpose was to get crystals that follows the original structure of chemical species. Here you can see there are no twins or any other impurities on our crystals. And then we made some research, and we know that, for example, in solar panels, there are crystals, and everyone is not aware of that and don't really know how does it works. So it was a base for us to know how things all around us works with crystals, and what are their application in industry, for example.
Can you maybe give us some examples of using this research in manufacturing applications?
For example, in the solar panels, it's silicium crystals that keeps the solar energy and send it to the electrical circuits in the solar panels. That's one of the examples that we found based on crystals.
Okay, thank you for your answers.
Thank you.
Any more questions? Anyway, we have to wait for our next participant from Spain. He hasn't connected yet.
we have time, maybe you can answer one more question. The crystal on the right, the left photo, the right crystal, how much time did it take to grow such big crystal up to 6 centimeters?
Can you see the mouse of
Yes,
that I'm using?
this one.
For those one, with an average concentration, it took like a week to get those results. And for little ones, it takes a couple hours. But it's very different from the concentration of the solution that we are using. For example, we have a crystal that is in a room for now a couple months, and it still around 5 centimeters. So it really depends on how do we concentrate our solutions, and it affects the growing time.
Okay, super.
And the right one, crystal, is it monocrystal?
Is it a monocrystal?
No, with a scale. The left picture, the right crystal.
I don't understand the question.
The crystal, which is on the left picture.
Yes.
On the right side of this picture, the biggest one.
Yes.
Right. Yes. Is it monocrystal?
Yes, you don't have any twins. Like I'm gonna find the picture. When they are twin, crystals are looking kind of like this. And when they are growing up, they still have this shape. And so when we obtain the results that you've seen just before, there are no other crystal linking to it or crossing it. So the crystal that you were asking me if it was a monocrystal, it was.
Okay, yes. Thank you.
Tristan, as far as we are waiting for the next participant, do you have some plans to grow crystals from another materials?
Like other chemical species, or?
Yes. And which one?
We've tried copper sulfate, which gives us the blue crystals. The dihydrophosphate gives us those crystals, and we've tried also with salt. It's very basic, but it's really hard to have big crystals, big monocrystal of salt. So we are still trying with salt. We also tried potassium alum. I don't know if it's a good traduction in English, but we also tried to use gallium to make it crystallize, because it has a really low fusion point, and so it's really easy to make it crystallize around crystals, for example.
Okay, thank you.
Maybe we can listen to another participant
In
For
5 minutes,
example,
there should be our participants from Italy connected. We have our participant from Slovenia, but I think if we start listening to him now, our timetable will go, will crash, I think.
just change them.
I think we should wait either for participant from Spain or for participant from Italy. If the participant even from Italy will not appear, we will hear to our Slovenian participant.
Okay.
And now we have our participant from Spain. Hello, Jorge.
Hello, can you hear me?
Yes, we can.
Okay.
I'll remind you about our time limitations, 15 minutes on all, and for presentation and for questions.
Okay. So, should I start?
Yes, you can.
So, hello, my name is Jorge. I come from Spain, and today I'll be speaking about my project about how I characterize the variable star EPIC 246-257-206 discovered with data from the Kepler Space Telescope. So, the main research objectives were to find the variable star in the Kepler Space Telescope database, understand why the brightness of the star varied over time, and to do that, you need to calculate the period and the amplitude, and finally, to publish these results in the International Variable Star Index. Before that, I just want to mention some brief concepts that are needed to understand the experimental process and the conclusions of the research project. The first concept that I have prepared is the concept of a light curve, where it's a plot that in the y-axis, we've got the brightness of the star in units of magnitude. It is an inverse scale. And in the x-axis, we've got the brightness of the star in units of Julian date. If the star is variable, we can see that the observations do not line up in a horizontal line parallel to the x-axis, but they will oscillate through time. And most of these variable stars are periodic, meaning that this oscillation, this pattern that eventually will tell us why that star varies, will repeat over time. And if we do not have enough observations to see this pattern nicely, we can superimpose all cycles, all observations, and obtain a new plot called the phase plot. This plot will tell us exactly at which moment in the cycle was that observation taken. And for that, we need to define a new magnitude called phase, which is the integer, which mathematically is defined as the integer, as the decimal part, sorry, this was a mistake, of this quotient, which is the time of the observation was taken minus the initial time of the cycle divided by the total period of the cycle. According to the general catalog of variable stars, we can define 5 different groups of variable stars, and more in particular, eclipsing variable stars consist of binary systems where 2 stars orbit periodically around their common center of mass. When the big star eclipses the small star, we're going to see a primary eclipse, so a primary minimum in the light curve. And when the second star orbits, eclipses the big star, we're going to see a secondary eclipse in the light curve. Using this idea of the light curve consisting of 2 eclipses, we can define three different types of eclipsing variables. The first type is the EA type, which consists of very well-defined eclipses, and outside those eclipses, there is no variability. In the EB types of eclipsing variables, the eclipses are still well-defined, but there is some sort of variability outside those eclipses. And finally, we've got the third type, which is in constant change of brightness, which is the EW type. Quickly, I just want to mention the concept of parallax that is mainly used in astronomy to calculate distances to nearby stars in our galaxy. So when we observe a star in January, we see that it has a position relative to some background stars. And when we observe it 6 months later in the other side of the sun, it has a different position regarding the background stars. Comparing these images, we can obtain this angle here that using some simple trigonometry, we can define the distance of the Earth to the star as one astronomical unit divided by the tangent of the angle. As I said, I used the Kepler Space Telescope to find a variable star. And I used this because it used photometric data, that means brightness over time, and its main focus was to find exoplanets, and to do that, it used the transit method. It studied the photometry of the star, and it looked for depth in the brightness of the star. That means that a planet has gone in front, similarly as in the eclipsing variables, producing a depth in brightness and an eclipse. Like its photometric observations, brightness over time, and we've got a cadence of every half an hour, we've got a data point. It was a perfect way to get incredible amounts of data to find a variable star. More in detail, I decided to mine the database in the second part of the mission, in the K2 mission. And more in detail, I used the field of view of campaign 12, which was centered in the constellation of Aquarius, which is favorable to see in the winter months when this observation was taken. In this campaign, I used a process called data mining, and I only looked at those stars that were bright enough to be observed from Earth and with the available equipment. The star that I was looking for had not to be cataloged, it had to be a variable star. To do that, I checked the Vizier database constantly, and also the period of the star had not to be longer than one night to then observe it from Earth, to not have to see it in more than one night to obtain enough data to characterize it in one night. Then in this campaign, I found the star EPIC 246-257-206, which from data from the Gaia spacecraft, I obtained its parallax angle and found that it was about 700 light years away from Earth. Later, I had to check that the Kepler photometry was valid because I wasn't responsible of obtaining this data. So then I used the Pansters telescope in Hawaii, which provided me this image, which is high definition, which is very high definition, which covers a greater field of view of the Kepler image, which is this one here. These pixels are very big in size, relatively big, and there might be a faint star that in this constant doesn't appear in the mask, but it does appear in our data. Therefore, using this data from Kepler, you can say from Pansters, we can say that there is no fainter star in the field, and therefore the data from Kepler is valid. Further on, I observed it from the ICTA observatory in northern Spain, where I obtained images like this one, which using a process called aperture photometry, I compared the flux arriving to the variable star in question with two other reference or calibration stars. Knowing the magnitudes of the brightness of these two stars, I can obtain the magnitude, which is the brightness of the variable star. Having all this data, and so the K2 data, the data from the ICTA observatory, and the data that Faustino Garcia, an astronomer from the Omega Astronomical Society, provided, I could obtain the light curve. And using a mathematical tool called the Lomsdorff periodogram, I looked for periodic signals in our data, we can see I obtained this graph, which for every possible period, it obtained how powerful is that signal. We can see that there are two peaks. The first peak corresponds to every time which is more frequent than the second one, corresponds to every time there is one eclipse in the light curve. And the second one corresponds to every time there are two eclipses in the light curve, which by definition is the period of a variable star. So using this, because this peak is double the size of this peak, I mean in period. So having this period calculated, I was able to obtain the phase plot, which allowed me to obtain the pattern that we're looking for, that will eventually tell us that it consists of an E8 type, because comparing it to the light curve at E8 type, we can see that this star is an E8 type. So what's causing the variability of star is that there's consists of a binary system that they orbit periodically around their center of mass, producing eclipses as seen from Earth. But most particular, that most interestingly is the amplitude of the star of only 0.05 magnitudes. And I suggest that this is because they're grazing eclipses. So in the plane that we are observing it from Earth, the stars do not eclipse totally, but partially, meaning that not all their surface is being blocked by the other star, and the shadow that's projected to Earth, the eclipse, is much smaller. Anyway, this was enough to publish the data in the variable star index, which was reviewed by the moderators of the catalog, and they agreed with my conclusions. And also, I just want to say how the importance of this project and its contribution to the search of an Earth 2.0, so to other planets similar to Earth. When we find a transit depth at a transit, we have these three transits here, for example, which are more U-shaped. But grazing eclipses in eclipsing binaries present also very low amplitudes in brightness, so very small depths, but they generally are more V-shaped. But this is a very small difference, and computers do not normally find this easily. So it is essential that we find the whole population of these stars in our galaxy, so we can take them off of the possible transit exoplanet candidate lists. And briefly, just want to say that this project allowed me to characterize and find other variable stars, as we have here. And thank you very much for your attention, to all institutions that helped in this project, and for this committee to allow me to present my project today.
Thank you for your presentation. We have a time for one brief question, and we will move on.
Jorge, thank you for your presentation. So you actually discovered a new star for a catalog, yes?
Yes, I'm being recognized as the discoverer, yes.
That's very cool, I congratulate you with that. And did you have access to observatory yet?
Yes, I had access to the observatory of this institute, of the ICTA Institute, which is part of the University of Oviedo in northern Spain.
Okay, thank you for your answers and report.
Thank you.
Okay, thank you for the question. Unfortunately, we do not have time for more questions. We will move on to our participants from Italy. Elvis? Thank you for your presentation. Elvis Barbeio. Correct.
Yes, good morning.
It's your turn. You can start your report.
Yes, I'm with my colleagues. I'm waiting them.
Elvis Barbeio.
Yes, good morning.
It's your turn. You can start.
Yes, I'm with my colleagues. I'm waiting them. Okay, I share my screen. Can you see my presentation?
Yes, we can see.
Okay. Good morning to everyone. We are Alice Barberio, Mattia Brollo, and Andrei Grase from Istituto Superiore Ascanio Sobrero in Casale Monferrato, Italy. And the name of our project is the Circular Economy of Rice, from Processing Waste to Bioplastics, BioNCS. So nowadays, the production of material used for food packaging focuses on sustainability and on environmental impact. So the main purpose of our project is to synthesize a bio-based compound material made of starch and nanocellulose, entirely obtained from the waste of rice processing. So as you can see in the image, we have from left to right, the broken rice, paddy rice, and the rice husk. The first step of the synthesis of the biopolymer was the extraction of the starch from paddy rice, which has resulted to be the best kind of rice waste. Then nanocellulose was extracted from husk. The BioNCS film was produced from a mixture of starch and nanocellulose. The two components present were combined in different ratios to evaluate their effect on the chemical properties of the compound. The most innovative part of our project consists in the extraction and the use of cellulose nanocrystals obtained from husk. Moreover, we can say that husk is no longer considered as a waste, but as a biomass used for the production of pellets, used, for example, as a fuel material for stoves. Our proposal would like to be an efficient ecological alternative that enhances the high amount of cellulose in the husk composition. We would exploit the advantage of this nanocellulose to improve the chemical, physical, and the mechanical properties of the compound. In the image is shown the biofilm we produced. Now, Mattia will explain in particular processes of extraction and the characterization of starch and nanocellulose.
Okay. Hi, I am Mattia. Of course, to extract starch from the various types of rice, we had to follow precise steps, which are shown in this image. We had to destroy rice into extremely small pieces so that the result of extraction would be better. The most important thing is to sunder starch from other various elements found in rice, such as proteins and other residues. The next step was to characterize the so extracted starch, which means to establish its qualitative properties. The first test we've done was the Lugol assay to assure that we succeeded with the extraction process. From this image, you can see that the blue-colored solution is indeed starch. On second place, we used a polarimeter, which is shown in the picture down on the right, to determine the purity percentages of starch obtained from broken rice and paddy rice. The former was pure at 100%. The latter was pure just for 84%. Going on, we had to extract nanocellulose too, because we found it enhances the properties the BioNCS film could benefit from, such as windability and flexibility, thanks to nanocellulose nature. We proposed an example of industrial chemical plant to extract it, shown in the picture on the left. The process we paid most attention for was the bleaching process. It brings better results using hypochlorite. However, due to our interest in minimizing environmental impact, we used oxygenated water, which gives a slightly less impressive outcome. As for rice, the next course of action was to characterize nanocellulose, giving it a look under optical microscope. In these pictures, cellulose nanocrystals can be observed, obtained looking at it with various lenses. This is, of course, important as it gave us assurance what we've got was indeed nanocellulose. We will next examine the obtainment of the BioNCS film and its properties in the coming slides. Andrei will explain us the process.
Hi. So to synthesize our biopolymer, we first solubilize the starch with an acid hydrolysis. Then we added the nanocellulose solution. The formed mixture was then laid in a petri dish that will let rest until the water was evaporated. We analyzed the windability of the polymer by bending it on a metal cylinder, and we noticed that the film with less starch was less windable than the one with more starch. Then, thanks to the IIR analysis, we determined the presence of starch in nanocellulose in our biopolymer, as we can see from the spectra. And we can also confirm that both sides of the polymer are homogeneous. From the TGA spectra, we can see the degradation temperature of the film and its components. Also, we can see many similarities with our DMA spectra, with the spectra of other materials used for the packaging of aliments and such. So, in conclusion, we created our project with a view to circular economy by exploiting the wasted crops of our area. We find out that the most suitable starch for synthesis of the compost was the one extracted from paddy rice, and the biodegradability test of our biopolymer confirmed this statement. Among our future goals is to replace the acid hydrolysis with enzymatic hydrolysis for a more eco-friendly alternative. Thanks for your attention, and if you have any question, please let us know.
Thank you for your presentation. Any questions?
Thank you for, yes, I have a question. Thank you for your report. You tell us about the economy. Can you please describe it in more details?
Yes, so I said this circular economy that we try to improve. So in our area, we sell a lot of rice and such, and many wastes are produced. So we try to make a gain by using these wastes of the production of rice to create a new material to sell. So that's what.
Yes, and doing so, we tried to exploit the many advantages these products give, because yes, they are used. They are not used because they are a waste, but we found out that they have many possible uses, and we try to give them one by doing this project. Or at least we tried.
Okay, I see. Thank you for the answer.
Any more questions? Curiosities?
And about these waste products, maybe it will be better to make organic fertilizers from them.
Yes, they ask from the rice that we used to make the nanocellulose is already used to make this kind of products, but we decided to use it another way. So to create this biopolymer.
Because you should maintain the organic status of the soil where you grow your rice. So you should just all your residues should go back to the soil, just to maintain the amount of organic matter in it, isn't it?
Yes, but we do use other kinds of materials that they have the same final result. Such we in this zone we have a high. Yes, we do have a lot of cows, and so their excrements can be used as a very good way to try to maintain the soil fertile, and so we try also not to use many chemical products, so we can use the husk and the rice, not only because to this kind of use, but we can use it to do something such as plastic, which is a big issue in our society and in our time. And so maybe this could be a valid alternative.
Thank you.
Thank you for your presentation. Thank you for your questions. We have to move on to our next. To our next participant, Luka Irinej Pichan from Slovenia. Thank you for your presentation.
Thank you.
Thank you.
Have a nice day.
Thank you so much.
Hello, everyone.
Hello, we can hear you.
Okay, I will now share my presentation. Can you see it?
Yep, we can see.
Okay, thank you. So I'm going to present the interdisciplinary research from our research project from Slovenian delegation. So I am Luka Irinej Pichan, student researcher, and I started this work in August 2017 as a high school student at the Biotechnical Education Center and completed it in September 2020. With the help of my mentor, supervisor, and coordinator of the research work, Marko Jeran, from our Faculty of Health Sciences, Faculty of Electrical Engineering of University of Ljubljana. So the main goal of the research was to determine the chemical and microbiological profiles of 10 varieties of cannabis, to find out the concentration of phytocannabinoids in each variety, and to check how the concentration of the same variety changes in 2 different growing seasons. The purpose was to check the microbiological quality of dried cannabis samples after the storage phase, and determine how the quality itself is affected by growth and storage factors. In order to determine the safety of every single sample that could be used for food purposes, we also determine the presence of mycotoxins, or more precisely, aflatoxins, in individual samples. So cannabis, commonly, or cannabis sativa, commonly known as marijuana, has been used by mankind since ancient times. For more than a millennium to the end of the 19th century, it was among the most important agricultural crops and industrial raw material. The vast majority of fibers and fabrics, like oil, paper, and medicines, were made from hemp. Nowadays, hemp is one of the most chemically researched plants, along with more than 120 known compounds. The most interesting ingredients are found in the secretion glands called trichomes, which are distributed over the surface of hemp plant. Although trichomes are found all over the plant, or the entire surface of male and female plants, they are especially concentrated in some parts of the female inflorescence. The resin secreted by trichomes contains various ingredients, among which cannabinoids, terpenes, and flavonoids are the important ones, which are secondary metabolites or secondary products. Cannabinoids are actually the main bioactive substance in hemp. Literature reports over 90 different cannabinoids that have been detected until today, although some of them are degradation products. Cannabinoids are, in broader sense, substances that bind to cannabidiol receptors and cause certain effects through this binding. The best known of all are CBD and the psychoactive THC. So our research was actually divided on 3 different parts, and I will discuss the hypothesis for those 3 different research areas: quantitative determination of cannabinoid concentration, microbiological quality of the samples, and determination of mycotoxin concentration in the section of discussion and results for each part. So the purpose of our microbiological part or analysis was to examine the microbiological quality of individual leaf sample of different varieties of hemp grown in 2017 season after 2 years of storage in a closed dark place with an average room temperature of 20.5 degrees Celsius and relative humidity of 39%. The results showed that all samples contained different amounts of microorganisms, which is most likely due to the design and characteristics of each of the variety and its resistance to pathogens. Samples were stored in food storage bag with a closure system and appropriately labeled. Microbiological quality was checked according to the guidelines for microbiological food safety, which are intended for the final consumer. Samples of dried cannabis leaves could be used as a herbal infusion. So we used guidelines to determine the permitted limit value of individual microorganisms for tea, coffee, and similar products. As seen from the standards, none of the samples exceeded in the permitted limit values, which indicates good manufacturing practice, appropriate hygiene standard, and appropriate storage. It can be concluded that the microbiological quality depends mainly on the individual variety and its resistance and adaptability to growing condition. But in any case, there could be a contamination of the sample during the treatment. This factor cannot be ruled out as a possible source of plant contaminants, as soil quality, soil moisture, soil microbial flora, humidity, air temperature, and the number of sunny and rainy days significantly affect plant health and consequently the presence of microorganisms. Here we can see some pictures from lab, from work. Next part was chemical part. So the chemical part was actually to determine the concentration of cannabinoids in dried buds of hemp from 2 different growing seasons. And we did that by high performance liquid chromatography, or HPLC, after 2 years of storage in the actually same space as for microbiological analysis. We found that the only variety of 10 different varieties was Futura 75, had a total concentration of cannabidiol above the variety of specification, while its concentration in the remaining 9 varieties from both seasons was below the lower limit of the specification for each variety. The measured tetrahydrocannabinol contents ranged from 0.01% in the use of 31 variety to the 0.63% in the Antal variety. All samples had a total concentration of tetrahydrocannabinol below the permissible limit of 0.2%, except for the Tisa and Antal varieties. The main reason for the difference of concentration in 2 different growing seasons that we compared was the earlier collection of cannabis samples, so 26 days in 2018 compared to the 2017, which also meant a shorter vegetation cycle and a cessation of growth during the period when the plant produces the most cannabinoids. Our last part was focused on the concentration of mycotoxins. So we determined a certain quantity of mycotoxins in most samples, but this amount is generally below the statutory limit. In addition to previously performed microbiological analysis, we also performed morphological identification of molds that had grown on the media using imaging kit. We found the presence of molds Fusarium, Cladosporium, Alternaria, and Rhodotorula. None of these molds excrete large amounts or do not excrete aflatoxins at all, which is also evident from our results of measurements of the presence of alpha toxin. Unfavorable growing conditions, improper handling of plant material and storage cause the growth of molds that release aflatoxins. During storage, moisture is one of the key factors influencing the quality of the sample, as it creates a favorable environment for mold growth, so it has to be monitored at regular intervals. In conclusion, we found that in the same varieties growing in two different seasons, a difference in cannabinoid concentration occurred. One of the key factors influencing this difference was the shorter vegetation cycle, which meant that the phytocannabinoid formation and excretion phase was not completed. Based on the treatment of samples, quality, growing condition, and appropriate drying and storage, was anticipated that the amount of microorganisms present will be less than specified by the standard listed in the guidelines. The samples were dried and stored for a long time, which creates quite unfavorable conditions for the growth and reproduction of microorganisms. We can conclude that we handled the samples appropriately throughout the process and that they would be appropriate and safe for the end consumer. We anticipated that a higher presence of mycotoxins would also be detected in those samples in which microbiological analysis would show a higher presence of the mold and yeast. A higher amount of mold was detected mainly in the variety USO31. When the analysis of the concentration of aflatoxin in the plant material was detected, we did not detect them. Therefore, the last hypothesis is the only that we did not confirm. Mycotoxins are usually released into the environment by molds of the genus Aspergillus, but these have not been identified in any of the samples. And consequently, the concentration of aflatoxin is very low. The plant analyzed material is both microbiologically and chemically safe for use in human nutrition. I would also like to thank everyone who helped me with my research work, especially mentor and commenters, and to you who listened to my presentation today. Thank you. And if there are any questions, I'm glad to answer them.
Yes, I have a question. You said your examples are safe for the final consumer. Maybe, I'm sorry, I have missed something, but can you repeat, please, what is the application of your samples?
Sorry, what is the?
What is the application of your samples? How can you use it?
Yeah, so our samples could be used to for let's say for as example a tea, and they also could be used to make or isolate other bioactive compounds and used as a treatment, as a medical cannabis treatment actually. So our goal was to make a profile of 10 different varieties and to see which one of them is the best to grow and which one of them has the best profile and can be further used in any way that somebody could desire, because it's a raw plant material that can be used in industry, medicine, or at, let's say, at home.
I see. Thank you for the answer.
And can you tell us your own contribution to this work? Because it is a lot of material, and we want to know what are your.
So I've been working on this project for nearly 3 years in cooperation with university and governmental project. So in my work, actually, all the presented work is actually my work, and with help of mentors and supervisors, but I participated and I did all the analysis of 10 different samples. So from the result to the discussion, it's all my work actually.
Great, thank you.
Thank you for your presentations. I can see no more questions. And I propose to move to our next participant, João Carvalho from Portugal. Thank you. Hello?
Hi. We will just share our screen right now, if we can.
Yeah, you can.
Thank you. No. So we would just like to thank you for this opportunity for us to share our work. I'm Sara, this is João, and this is Clara here. So Atmos intended to quantify the ecosystem services provided by living green walls for atmospheric pollutants removal in urban areas, such as Porto, which is the city where we live. The project was developed during 2 years, along which we established 6 major work packages corresponding to our project lifecycle. The work package 2 and 3 and the exploratory study occurred mainly during the first year. This exploratory study was basically the direct application of conventional methodologies for quantifying particulate matter in bioabsorbers, using area function for calculation. We found it was not too accurate as PM penetrates through the tree canopy, so we developed a new methodology which we called Atmos methodology, based on volume function for PM calculation. This corresponds to our experimental study. Atmos is really an interdisciplinary challenge. Through a simple mind map, we could establish several key concepts that guided our research, from urban environment specificity to atmospheric pollutants characterization and PM health-related problems. We reached nature-based solutions, such as living green walls that provide several ecosystem services as PM sinkers. Describing it from a scientific point of view is fundamental, but not enough. So we tried an economic approach in order to quantify these ecosystem services provided by nature. This way we can establish a dialogue with municipality and give arguments to a real implementation of Atmos outputs. With this in mind, we established 3 main goals. 1, quantify PM retention along a highway near our school. 2, validate a new methodology to calculate PM retention in trees based on the volume function. And 3, assess the economic value of the ecosystem service provided by Atmos trees. These pictures here are from Atmos trees, where you can see the PM material adsorbed to the surface. Why this location? Firstly, this map represents Porto, approximately 50 square kilometers, limited by the Douro River, the Atlantic Ocean, and the cities nearby. From the yellow triangle, we can see that the sampling area is located in one of the most degraded zones in terms of air quality. This is mainly due to it being one of the main exits and entrances to the city. The school playground overlooks this highway. If we think children ventilate twice an adult, we can understand the importance of studying measures to minimize the impact of PM emissions. So we established a group of 10 trees along 150 meters in the highway to do our research. Understanding air dynamics is crucial for the study of PM retention in trees. Our trees may potentially capture PM from the main road and from the two secondary roads. The buildings may act as dispersion barriers. A future research would be to control all these variables in the study. So how did we quantify the PM in the leaf surface? In order to proceed a leaf scale PM gravimetric analysis, we collected 300 grams of leaves from 10 Lawson cypresses from the 4 cardinal points. Each sample was divided into 3 subsamples of 100 grams, 1 as control and 2 others for processing. PM retention was determined by measuring the area of the unit sample, that is the area occupied by each 100-gram sample. After scanning the unit sample, CAD software was used for the area calculation. Atmos introduced a volume function for calculating PM retention at a tree scale. This calculation assumes PM is not only absorbed to the surface, but also by the innermost foliage of the tree. By a trigonometric method and making use of the photograph of each tree, as well as the diameter at breast height measurement, we determine a value of the volume for each tree. The inner part of each tree corresponding to the trunk and the branches without vegetation was removed from the volume to be considered for PM retention calculation. To calculate the total amount of PM absorbed in a tree, it was necessary to estimate the bioabsorbent area of the tree. This was made by geometrically reducing 1 cubic decimeter of leaves into an equivalent area using CAD software and scanning. This table summarizes our calculations. If we use a conventional methodology to calculate the services provided by our trees in PM retention, we obtain a median value of 12.6 grams per tree. If we use the Atmos methodology for the calculation, the median value reaches 3,720 grams per tree, about 295% higher. So the following equation was applied to calculate PM retention by unit sample. We used twice the leaf area, assuming that PM absorbed in the both surfaces. Of course, we can introduce an error because particle deposition may not be the same on both sides of the leaf. Now, scaling for the tree retention, Lawson cypresses have a conical shape. So the volume calculation made use of the trigonometric formula for conical shapes. A correction factor of 50% was applied to remove blank spaces between leaves, assuming this percentage as the absorbent surface in the tree. The following equation was applied to calculate PM retention in trees using the volume function. If we use conventional area function to PM tree dust retention instead, the formula simply adjusts the unit leaf area to tree area. Finally, the FTIR spectroscopy provided us with information on the major functional groups present in Atmos samples. 10 FTIR spectra were acquired from the samples. The organic fraction of the residues have interesting structural characteristics compatible with the presence of aliphatic structures, aromatic structures, as well as the presence of hydroxyl and carboxyl functional groups in the protonated form. The economic approach was made by comparison with reference values from the same area from another study, which is called Futuro, that uses deciduous trees instead of evergreen trees. In the Futuro study, 8,167 trees were planted in several biospots along 4.7 acres of the highway we are studying. Research indicates that evergreen resinous trees have more absorbent capacity than deciduous trees. This reveals the importance of conducting a proper cost-benefit analysis regarding choice of the biomodel for the purpose of PM retention prior to the intervention on urban spaces. The value of PM 2.5 retention obtained in Atmos evergreen trees was 148.82 kilograms. The economic value of the ecosystem service provided is 1,107 euros. We didn't develop a temporal research, so we cannot assume that value per year. But if we look solely to Futuro variables, we can see that 1,167 deciduous trees capture 16 kilograms per year with an economic return of 119 euros. If the Atmos methodology of PM retention calculation was used instead of a conventional area, the values would be very different and more realistic. The PM 2.5 retention would be 15,000 kilograms per year, and the surfaces provided in terms of economical value would reach 111,645,000 euros per year. So finally, for further research, we would like to develop a PM deposition rate study by understanding the tree saturation point, calculating the efficiency of the living green wall, and by calculating how many trees are necessary to offset the emissions of this urban area. Furthermore, by using iTreeStratum software, we would produce more accurate data by using the growth curves of the Atmos tree species and by calibrating this model with the geographic and economic data from Porto, like the climate, buildings, air pollutant concentrations, and rainfall patterns. Thank you for your attention, and if you have any questions, we are available to answer.
Thank you for your presentation, Sergey.
Yes, thanks for presentation. Are you the only one group who provides this project, or there are several groups in your city?
So in this group, in our work, we are the only ones who have this project, and the other data are from other projects like the Futuro we mentioned, but they don't use the same trees nor the same method, because the method is ours. We invented using the volume instead of the area, but we use their data because they're in the same area, so we make some extrapolations for the economics and things like that.
Okay, and did you actually grow these trees across the streets, or did you actually grow this?
No. We used the trees that were already there in the highway. That's why we said we couldn't make the temporal research, because we don't know how long those trees have been there. So we would have to make another study to know the exact capacity of each tree and how much they can absorb.
Okay, thank you very much for your answers and report.
Thank you.
Okay, we have time for one more question. Anyone? So I think we can move to our next participant. Thank you for your presentation.
Thank you.
Thank you.
Asia Zia, if I spelled it correctly, it's your turn. Asia. Okay. We can see you.
Good afternoon, judges, panelists, and fellow scientists. My name is Asia Sial, and I will be telling you about my project called the Magnimal. And I will be presenting a project and hopefully something that will avail in the future. Edward Teller said, "The science today is the technology tomorrow." I will be putting this quote to use by expressing my project. What is mankind's greatest invention? Energy. We use energy in forms of electricity to cook, to operating appliances, computers, electronics, lighting, and refrigeration, to name a few. But with every seed of good is always a seed of bad. And with energy, we have produced our prime energy source, fossil fuels. I will be trying to tell you the bad and the good of our energy sources, and I will present to you a presentation telling you about my project. Can everyone see?
Yes.
The question, how can we generate cost-efficient, clean energy with places with low resources? This has been a problem in many countries and continents, including my own country, which is Zimbabwe. Some places, especially near the equator and the two poles, have low resources and low prime winds. So energy is very scarce, and the alternative, which is fossil fuels, damages the ozonosphere. So proposing a new energy source would increase the chances of living a cleaner and safer life. My hypothesis is that if we introduce a new alternative energy resource, it would clean and change the world. And by changing the world, we would rid of all the bad gases and bad gases that destroy the ozonosphere and isothermal layer. If the isothermal layer is being damaged by fossil fuels every day, this idea could resolve it and therefore decrease global warming. This is a cheap and ubiquitous, and the worldwide problem will be eliminated by more than 68% if this energy resource is used. My energy resource uses two simple methods: magnetism, the forces of magnets from the two poles, and oscillation, which is the movement of an object that swings inward. The purpose of this project is to eliminate world problems. And by creating a whole new renewable energy resource using magnets, the magnetic force and oscillating force, we could produce clean electricity. This has been a really big problem, and clean electricity is really hard to get. People use solar, wind, and geothermal type of energies, but places with low winds, places with less sunlight, such as near the poles, such as near the equator, people can't use this. And it's also very expensive, costing up to thousands of dollars. And small villages will not be able to afford these type of inventions. So solar and wind is a very good idea, but a very low resource. And this idea is to use the windmill design, using magnetism, oscillating force, to create a whole new energy resource that will be very cheap to get and very easy to make. The oscillating force is a movement of a back and regular rhythm. This is my design of my project. There will be an oscillating magnet that will repel the magnet on a propeller of a designed windmill. The magnets will therefore repel on each of the windmill propellers, which will cause the drive shaft to rotate, and the generator will therefore create electricity using the high-speed shaft. This method can be used in places where the wind is very low, because the magnets will therefore do all the work. And this design will be able to last for many years to come. My methodology: I made two prototypes to test which one could work better. I collected recyclable materials to make my prototype. I used the PC fan of an old computer to make a prototype of how the windmill will work. This is a picture of my first prototype. As you can see, contains only 2 magnets on the side. And the second prototype contains magnets on all the propellers. As I added the magnets and did a test run, the second prototype did not function as well, and the results were very poor, causing the bulb that I tried to experiment dim. The second prototype worked flawlessly, as the energy on one side is being reused all over again, and this created the light bulb to light for many hours as I tried to do this experiment. I tested it again and the results improved as the energy was sufficient to light the LED light. With this experiment, I tried to use this method to light a bulb in my room, which lit for many hours, and through the night, it lit as a night light. If we had to create a huge big one, it would have enough power to power an entire village. This chart represents the difference between my first and second prototype. It shows how the first prototype, which is blue, worked faster than the second prototype, which is in red, works slower. The blue prototype had much more efficient results, from 15 rotations per second, but the red one had 30 rotations per minute. The effects worked because one prototype had the energy going through on the other side, and it was uneven, causing the propellers to go faster. The second prototype did not work as well because the energy was all even and it would not repel as it was supposed to be. I conclude that the Magnamill is a very functional invention with a motor to produce energy to use in the poor areas and regions with little wind or little sunlight. The design is similar to a windmill, but we would use magnets that would repel and rotate the propellers. My hypothesis was supported by my results correctly. If you have any questions, please feel free to ask.
Thank you for your presentation. Questions, your experts?
Thank you for your presentation, but can you tell us again what the principle of your device, how it works?
It works using magnets. So say an oscillating magnet would be repelling the 2 magnets on a propeller. The propeller would then be rotating because 2 magnets on one side would be repelling each other. And if a magnet repels each other, it causes movement, it causes energy, and it is a type of energy that will enable the propellers to move. And through the oscillating magnet, it would repel it even further, and therefore it would make the propeller to work faster.
To be honest, it's not very clear for me. Maybe you can show us.
Show you a model?
Yes, can you?
Yes, I can. I have it right now. This is just a model, but it is not exactly the same thing. It is just a model to show how it would work.
And tell us why it will rotate. Why? How?
You see these magnets here?
Yes,
they both
yes.
have north and south poles. And if I had an oscillating magnet that would move like this, it would move like this.
Another magnet,
oscillating, it would repel these 2 magnets and it would cause this to rotate around.
Why another magnet will oscillate? How?
It's being repelled. So when a magnet repels, it will cause a force, and that force pushes the propellers to go round. The force will create a very powerful force, and that force will be used to power the propellers and then therefore power the generator.
Why will the second magnet repel and rotate your fan?
Second magnet helps by increasing the force, and since it is only on one propeller, this one propeller will help generate power by pushing all the other ones. And by doing so, this will create the energy and power to power the generator. The first prototype, which had all magnets all around the propellers, did not work because all the power was distributed evenly. It was distributed evenly because the magnets were all evenly put on each propeller. And that didn't work because it would not push the next propeller to go round.
Yes, I see, but can you please show us the second magnet that is in this construction?
Okay.
Okay, we can see this, but the question is how the second magnet will move.
Move in an oscillating movement.
And
so,
oscillating by what?
By a type of strength. This is what I'm trying to improve in my project. As I continue my project into higher levels, I will try to find out a better method than the oscillating movement, and therefore power the magnet.
Okay, thank you.
Any more questions for our participants? Anyone?
No more questions.
So it was the last participant on the day, and I think our meeting today comes to an end. Thank you all for attending.
Thank you.
Thank you.
Thanks and goodbye.